unite us all. We are grateful for your presence and looking forward to sharing the knowledge, ideas, and perspectives. Let's make the most of this opportunity and create meaningful connections. A day without prayer is a day without blessing, and a life without prayer is a life without power. To begin the auspicious occasion, may I invite you all to stand for invocation to invoke the Almighty and for the virtual lamp lighting session. Thank you so much, everyone gathered here and are requested to take their seats. I now cordially invite our head of the department, Dr. R. Vijayvahani ma'am, to present a welcome address.
Good morning, all. Am I audible? Okay. Very good morning to and warm greetings to one and all gathered here. I feel extremely glad and privileged to welcome everyone for this wonderful program where we have arranged an online workshop on decoding the science of nutrigenetics. I'm indeed honored to extend my warm welcome to each and everyone gathered here. I would like to welcome our Dean of New Age Science and Technology, Dr. V. Subhashini, and all my fellow staffs of Home Science. I, on behalf of PG Department of Home Science, Food Science, Nutrition and Dietetics, I would like to extend a special welcome to all our chief guests. First, I would like to mention Dr. Janani Tamilvanan, Director, Institute of Nutrigenetics, Muscat, and Dr. Ferdin Mary Sharmila, Scientist, C. Jipmer, Puducherry. A big thank you to all the speakers for accepting our request and gracing this occasion amidst your busy schedule. Thank you so much, Sharmila ma'am. We are indeed waiting to hear to your session. And I would like to express my gratitude towards all the PG staffs and other staffs who sincerely contributed to this event in order to make this event a successful one. Thank you so much.
Thank you so much, ma'am. I now request our beloved Dean of New Age Science and Technology, Dr. V. Subhashini ma'am, to address the gathering.
Good morning, everyone. And I'm Dr. V. Subhashini, Dean of New Age Sciences, and it's my pleasure to welcome you all for the Decoding the Science of Genetic workshop. And I welcome the guests too. Workshop is like will provide a platform for collaboration, knowledge sharing, and it fosters innovation, creativity, and problem solving. And the genetics is an emerging field which focuses on gene structure, genotype, and inheritance patterns. This nutrigenetics and nutrigenomics definitely plays a major role in individual health and disease condition. It is nothing but tailoring the dietary recommendations on individual's genetic profile. Hope this workshop can provide the basics in personalized nutrition recommendations and helps in targeted nutrition intervention. So once again, I wish the department to continue this workshop. Thank you.
Thank you so much, ma'am, for your delightful welcome address. The goal of education is the advancement of the knowledge and the dissemination of truth, said John F. Kennedy. It is on such occasions that we get opportunities to widen our knowledge and go further in the pursuit of learning. Without any further delay, let us go on to the most awaited session of the day. I request now Tanvira Jasmine, her first MSc FSND, to introduce our first speaker of the day, Dr. Fadina Mary Shamila ma'am, to the audience.
Good morning. Dr. Fadina Mary Shamila is a highly skilled and dedicated scientist with extensive experience in diagnostic virology, genetic research, and molecular biology. With a strong educational background, including a doctorate in genetics from Birla Institute of Technology and Science, Pilani, she has established herself as an expert in her field. Currently working as a scientist C at RVRDL JIPMER, Puducherry. MERM has proven track record in managing high-level diagnostic testing, conducting epidemiological studies, and developing innovative research methodologies. Her expertise includes molecular diagnostic tools, genetic panel development, and statistical analysis of gene variants. With numerous publications in reputed journals, MAM has demonstrated her commitment to advancing scientific knowledge and improving diagnostic practices. Her research interests include infectious diseases, virology, genetics, and molecular biology. As a seasoned professional, Dr. Fadina Mary has honed her skills in training and mentoring students, as well as in practical lab techniques and advanced genetic analysis. Her expertise and dedication make her a valuable asset to her organization and the scientific community and to us.
Thank you, Tanvira Jasmine. So, the topic for the first session is Fundamentals of Genetics. Now, I request Dr. Fadina Mary Sharmila ma'am to take off the session. The dais is yours, ma'am.
Thank you, ma'am. Thank you for the warm introduction. Good morning, one and all. It's a pleasure to present in front of such fresh minds the beautiful subject of genetics. I will deal with two sessions, basically. The first session will be on basics of genetics. Hope I'm audible.
Yes, ma'am, you're audible. Yes, ma'am, you're audible. Please continue.
Thank you, ma'am. I will just share my screen now. It is in slide share mode, ma'am.
Yes, ma'am. It is in slide share mode.
Thank you, ma'am. So I'll go ahead with the first session, that is basics of genetics. And I believe that all of you are from the nutritional background. So we'll touch upon what is the basic terminologies that are used in genetics. And then we'll proceed for a short quiz session. Then the second one will be more concentrating on epigenetics and how we process the sample for genetic screening. So once again, welcome you all. So the first slide, we are talking about the importance of genetics in various fields. So we have all faced the current pandemic, right? So all of you are aware of the various variants of virus SARS-CoV-2 that was circulating. So there was a first one virus, Delta virus, then it went on to Omicron and all those things. So that is, we were able to catch this virus very much earlier because of the powerful tool of genetics. And the scientists were able to predict that this kind of variant, that is small changes in the nucleic acid of the virus, is making it more prominent, like it is causing more severe disease or it is spreading much more faster. All these information were available for the public uh, service through the governments, helping the governments to take appropriate measures. So there are several ways genetics can help us, right from the diagnosis of rare diseases to prevention of chronic diseases like diabetes, myopia, obesity, hypertension, hyperlipidemia. All these are chronic disorders. So, and you know that it is coming up in within the family. So we are inheriting such chronic disorders as well. So if we find them out early by genetic testing, we will be able to prevent or delay the onset of these chronic disorders. And this will definitely lead to reduce in cost, healthcare cost. And we can customize the treatment, we can customize the diet, we can customize the exercise pattern for the particular individual depending upon the genetic information that they are carrying. So in this way, genetics has a wide application, and it includes in prenatal assessment to check for the risk for chronic diseases, studying ancestry, and then if you take and see the 23andMe, there is a company which will tell us from which ancestry we are belonging to, then customizing treatment, weight management, etc., etc. So coming to the basic definition of what we see in genetics, this is a very basic definition. One is genotype, another is the phenotype. So phenotype is something that we see outside or something that is measurable. So all these pictures that you see here are something that is seen outside. So that is the phenotype of an individual. And the measurable traits, say glucose value or the value of your lipids, LDL, HDL, etc. All those are also phenotype. They are hidden, but they are measurable, right? How it is expressed, these phenotypes, like one person is a tongue roller whereas the other person is not. So this is determined by our genetics or genes, or which is in the DNA, and that is called as a genotype. So phenotype is something that you see outside, whereas genotype is what is the information coded by the DNA. So moving on to DNA, this we would have learned in our early 10th or 12th biology classes, like DNA, where it is present. So it is present in all the nucleated cells. All nucleated cells have the DNA inside it. So when we see the DNA has, this is the karyotype of an individual, we have 22 pairs of autosomes and 2 pairs of gender chromosomes. So totally we have uh, 23 pairs of chromosomes. Okay. So the two gender chromosomes can be XY in case of male and XX in case of a female. Apart from this, this is the nuclear DNA that is present inside the nucleus. Apart from the nuclear DNA, we have something called as the mitochondrial DNA as well. So what are mitochondrial DNA? They are circular DNA present in the cytoplasm. Okay. So you can imagine an ovum, an egg, and it is in the haploid state. What we call the haploid state is it has only one half of the chromosome. So only 23 chromosomes are there. The sperm carries the next 23 chromosomes. So when they fuse together, they form the zygote, right? But when you see, the egg is the one which has the cytoplasm. The sperm brings only the nuclear DNA content. So the cytoplasm of the mother has this mitochondrial DNA which is passed on to all the children. Okay. Whereas the chromosome from the father, the 23 number from the father and the 23 from the mother in the nucleus fuse to form the nuclear DNA, that is the 23 pairs. And that is, we get half and half from the father, half from the mother. Why it is important to learn about mitochondrial DNA is if there is a change, a variation, or a mutation in mitochondrial DNA, then that disease is carried out to all the children, okay, from the mother to all the children. So, as I told you, we have 23 pairs of chromosomes and we are made up of half and half from paternal origin, half from the maternal origin. So this DNA, which is present inside the nucleus, is containing the instructions for the cell. So the DNA carries all the information for producing all the important proteins, enzymes, mRNAs, miRNAs, and all these information are there in the DNA. The entire DNA is not carrying that information. There are sections of the DNA which we term it as genes. Those genes are the actual positions which will get converted, that information is getting converted to protein or enzyme. And the changes in that gene is called as an allele. We'll see it explanation in the further coming slides. So how this DNA is arranged inside the cell? It is, as you all know, it is a double-stranded structure where we have four bases, A, T, G, C. And according to the base pairing rule, A always pairs with T, C pairs with G. So when they pair like this double-stranded DNA is then tightly wound around proteins called as histones. These orange or yellowish structures are proteins, which are histones, and the DNA is tightly wound around these histones, which is further stacked upon each other tightly to form the chromosome. That is how dense the chromosome is. So you have a double-stranded DNA, which is wound around histones, which are proteins, and then it forms the chromosome. So like this, we have 23 pairs of chromosomes. So histones are proteins which are positively charged and your DNA is negatively charged. That's why they are able to bind around these proteins and keep the DNA safe inside the nucleus, because there are a lot of enzymes in the cytoplasm which can lead to the degeneration of the DNA. So these DNA is protected in when it is structured in such a format and kept safely inside the nucleus. So as I told you, A pairs with T, G pairs with C. The bonding between A and T is double bond, whereas G and C is triple hydrogen bonds. And the backbone is the sugar phosphate is the backbone. Now coming to the genes, as I told you, not the entire DNA is carrying your information or which can be translated into a protein or an enzyme. There are sections of the DNA which are called as genes. In that gene, we have about less than 2% of the entire genome containing these genes. So you can imagine some 3 billion bases are there, ATGC, like that, you have some 3 billion bases, out of which less than 2% is actually carrying the information which is going to give you the instructions for the cell. So when you see the arrangement of the gene, it is always represented from 5' to 3', and you have a promoter region, then you have something called as the start codon, then you have exon, intron in an alternative fashion, and then comes the stop codon. Why the structure is important? It is common for all these genes. All the genes we have right now around some 35,000 genes known so far in human genome. So all these genes have a promoter region, and then you have the start codon, then you have the exon, intron, like that. This structure is maintained because, say for example, you're hungry. So when you're hungry, you know that ghrelin hormone is should be produced, right? So it is not always your gene is very active and it's producing ghrelin always. So only when a particular stimuli is there, then that region of the DNA is accessed, and that region alone slightly opens, and your signal goes and binds to the promoter region. Once it binds to the promoter region, then it will start the process. So we'll see in the coming slides what is the process. And so this is how your gene is arranged: exon, intron, like that. So when you consider a gene, or when you consider the genome and compare it to a library, you can consider the cell as the library and the books in it as the chromosome, and each chapter is a gene, and the letters in that particular chapter which make up those sentences, that is the nucleotide. So how this is getting converted? So this we would have learned in our biology. So just a revision of what we have learned. In the central dogma, it is the conversion of the information present in the DNA to the actionable component, which is your protein or hormone. So first is DNA transcription, next comes translation. So when we compare it with baking a cake, so we don't bring the entire book of cakes into the kitchen, right? You need only one recipe out of that book. So what we do is we copy it, or we take notes of it, and only that recipe alone we bring it to the kitchen. So when you compare the cookbook to a DNA, that copying process is called as transcription or photocopying, and that particular recipe is called as messenger RNA or mRNA. Remember, DNA is there inside the nucleus, and it cannot come outside into the cytoplasm because it will get destroyed. So what happens is that this transcription process happens inside the nucleus where it is safe. And as I told you, the trigger will go and the triggering molecules will bind to the promoter region. Once it binds to the promoter region, it will start the process of transcription. So it is like a signal, okay, start producing. Then what happens? It will convert that, or just to copy that information from DNA to the mRNA. So the exact information, ATGC, is being copied to the mRNA with only one slight change, wherein the thymine or the T in DNA is replaced by uracil in your RNA. Now, this is a short fragment because that particular gene alone is copied, right? It is a short fragment. This mRNA is now then matured, because when you copy, as I told you, there are exons, exons, and introns. So once the RNA has first copied the entire thing from the start codon to the stop codon, what happens? It exiles the introns alone. So the introns are cut and the exons are joined together. All this happens inside the nucleus itself. Now the mature RNA, mRNA is ready. This mature mRNA comes out into the cytoplasm. Once it comes out into the cytoplasm, so it has come now to the kitchen example. Here the ribosome and your tRNA is waiting. Now, once it comes out, the ribosome will sit on the mRNA and start reading every 3 bases. It will start to read. So every 3 base is a codon, which codes for a particular amino acid. So once it reads the codon, say example AUG, which is the start codon, what happens? It asks the tRNA, which is the kitchen hand here, to bring the particular amino acid, which is the sugar. So what happens? The particular amino acid is got, then first amino acid, then it moves to the next 3 codons, the second amino acid, third amino acid. Like that, the chain starts growing. So you remember start codon, there was a stop codon as well. Once the ribosome reads the stop codon, it will stop recruiting the amino acids. Now your entire protein is ready. Either it is functional inside the cell, or it is secreted into the bloodstream and goes to the location where it has to be functional. So this is the entire process by which the DNA information is converted to RNA and then to the protein as such. So this is the amino acid table. Okay. So as I told you, there is one start codon, AUG methionine, which is the start codon. And you have several other codons for all these amino acids. And you have three stop codons, UAA, UAG, and UGA. So if the ribosome reads any of these three stop codons, what will happen? It will stop the translation process. Now your protein is ready. It will undergo after that modification, secondary structure formation, tertiary structure formation, and then it is ready for its function. So say for example, this is a very short strand. So I just wanted to, the positions are changed slightly, sorry. So you can see here, 5 dash to 3 dash. This is one strand and this is the other strand, okay? So ATG, so that is the first codon. Once it reads ATG, what happens? Methionine is got. Next it reads ATC. ATC codes for isoleucine. So that enzyme is joined. The last is TAA, which is actually your UAA. So it codes for the stop codon and it stops here. So sorry for the misalignment. Okay, hope I was clear till now. Any doubts, please note them. We will have a discussion at the end of the session. Um, Now continuing with the alleles. So we saw what is DNA, how it is organized inside the cell, how it is getting, how the instructions from the DNA are getting transcribed and translated into a protein. Now coming to the concept of alleles. So what are alleles? So we go back to the Gregor Mendel's experiment, the father of genetics. So here he described that the parent genotype is TT, capital T, capital T, small t, small t. So when the pure bred were actually crossed, what happened? They get segregated in the gamete stage. And then they combine. So this combines with this combines with this, like that. So this is the F1 generation. When they saw the F1 generation, one of the parent was tall, one of the parent was short. But all the children or all the plants in the F1 generation were tall, which indicates that some component from the tall parent was passed on to all the kids. Okay. So here he has represented it as capital T representing tallness, and all the wherever capital T was present, the phenotype was tallness, whereas the genotype was either capital T small t or capital T capital T. When he crossed such heterozygous alleles, what happened? He saw that out of the four combinations, one combination, that is 25%, the plants were short. So this is the concept of alleles. So we have individual units, okay, which can be crossed during the fertilization, and then depending upon the phenotype that it carries, it will express the phenotype. Okay. So in case of this capital T, it was associated with tallness. Small t was associated with dwarfness. So whenever capital T was there, it showed the phenotype tallness. And whenever small t was there, it will show dwarfness only when two of them, that is when we receive both the alleles from father and mother as small t and small t, then only we can show that dwarfness phenotype. So by this we understand that one is dominant over the other. Okay, so tallness was dominant over dwarfness. So whenever only one single capital T is there itself, it will show as tallness. Why this is important is when we have certain alleles, when we associate them. So the scientists or the publishing, so a lot of publications are there. When we read such publications, we know that a certain allele is associated with a certain phenotype. So when we check for an individual's genetic composition and we find that allele there, then we associate, okay, if this allele is there, then this is the phenotype they might show. This is the genotype, then this is what they will express. So that's how we correlate the alleles. So coming to the next concept, that is now we have done with the construction of DNA, how it is, what are genes, and what are the different kinds of alleles that can be present in a gene. Now coming to changes in the DNA. So when we compare our entire genome, okay, that is from chromosome 1 to the last chromosome, when we compare it with other organisms, we see there is similarity. Okay, so you all know that our nearest ancestor is chimps. We share about 98% similarity with them. When we compare two individuals' DNA, say for example, I compare my DNA or genome with another person's genome. When we put them side by side and if you compare it, the entire genome, we are different only by 0.4%, just 0.4%. But how we are looking so unique is because of the combination of alleles that we have. Okay. So changes in DNA could be good or could be bad depending upon the phenotype they are associated with. The most common kind of variation that have been studied so far is the single nucleotide variation. Okay, there is a change in a single base. Say, for example, here there are three people, person 1, 2, and 3. And this is color represents the dark violet represents the chromosome from the mother's origin, and the slight greenish color is from the paternal origin. Okay, so say this is the fifth chromosome. At a particular position in the fifth chromosome, what happens? Person 1 has C at one position, that is in the mother's chromosome, and the father's chromosome carries T at that position. So these two are the alleles. Same location in a particular region, there are two different bases which are present. So this person is heterozygous. That is, the alleles at that position are different. So we call them as a heterozygous and we write it as CT. So it is the genotype. CT is the genotype for this person. When you compare with the second person, you find that both the father and the mother carried C at that particular position. So the genotype here is CC. And for the third person, it is T and T. Okay. So at a given position, okay, a person can have either be homozygous for one of the alleles or heterozygous. Okay. So some of the variations are present commonly in the population. Say, for example, you take some 100 individuals and check for a particular variation. And if that variation is present, say the previous example itself, we'll take C and T. C and T, if you check C, C is present in 80% of the population and T is present in 20% of the population. So it means that they are common, that is greater than 1%. Whereas some variations, when you see, even the single nucleotide variations is very, very, very rare. So those kind of variations are called as mutations. That is, usually their effect will be very, very high. That is, their phenotype will be very severe and is usually associated with a disease. In this case, cystic fibrosis, there is a variation in the chromosome 7, there is a variation, and that single change itself leads to this disorder of cystic fibrosis. Whereas in case of chronic disease, any chronic disease for that matter, be it diabetes, here they have shown coronary artery disease. So you can see variations is there throughout the genome. So when, as a scientist, when we study, we find that small variations in different regions of the chromosome. The single variation will not lead to coronary artery disease. But when these variations are there in different positions, the effect of that variation all added up together is what is making that person susceptible for coronary artery disease. Okay. So this is the difference between a mutation and a polymorphism. This is called as a polymorphism. Now we called as variance. And polymorphisms are usually present generally in the population. And it is the combination of different polymorphisms put together that leads to a disease. Whereas in a single variation, it is not like that. It is a single variation itself will lead to a particular disease. And moving from single variations, single nucleotide variation is the commonest one and is the most commonly studied one. Apart from that, we do have other variations as represented here. It could be deletion of a region, it could be duplication of a region, inversion, insertion, translocation, etc. You have to just you can hear about these terminologies. That's it, because they are very, very rare. And such kind of huge changes usually will result in spontaneous abortions, in the when the in the uterus itself, when the child is growing. So when the zygote starts dividing, and there is by some mistake, the division is not proper, and the replication of the DNA is not going on correctly. So that time this kind of changes will happen, and the dosage of the genes are very, very high. So the fetus cannot withstand it. So these are very rare in general, whereas polymorphisms are quite common. So when you compare, these are single nucleotide variations, which are very common. Then comes your insertion deletions, which are not that very common. That is small insertions, small deletions like that. 1, 2, 3 bases gets deleted. Sometimes 10 to 50 bases gets deleted or gets inserted. That is all small variations, and their occurrence is also very rare. Coming to structural variations, it's much, much, much more very rare scenario. So these are called structural variations. These are the different kind of variations present in the DNA. And when we see that variable portion, this is the variable portion, whereas the rest of the portion is the same. That's why I told we are only 0.4% different, because almost everything is the same. But that variation in this single nucleotide variations, that small, small deletions, mutations, all that contributes to the difference between two individuals. Now, since all those are rare, we will concentrate only on single nucleotide variations, because that is what is being used most commonly in a hospital scenario or in such kind of precision nutrigenetics or precision medicine. We usually work with the single nucleotide polymorphisms. Okay, so it can be present anywhere, right? So it can be within a gene, it can be outside a gene. Within the gene, it can be in the promoter region, it can be in the exon region, it can be in the intron region. So depending upon where it is present, it is going to affect the consequent process. Say it is there in the exonic region. The exon is the one which is going to be carried to the mRNA to form the protein, right? So in the exon region, what happens if these kind of variations are there, it might affect the amino acid that is produced. So in that case, what happens? So this is the normal DNA of a person. If you see here, it is CCT. CCT codon codes for proline. Okay. If there is a change from C to G, what happens? So what happens here? Instead of proline, it codes for arginine. So the amino acid is changed. This is substitution. Okay. And here it is insertion. Say for example, CCT. Instead of that, what happened? A G got inserted here. So this C is shifted here. C, G, C. So the frame itself is shifted. So what happens? That instead of proline, lysine, valine here, it goes to arginine, and then one stop codon comes in between. So it is not possible. This kind of variation is called as nonsense variation, because it leads to a premature stop codon. Your protein is not fully formed. It is halfway, and suddenly it stops the translation because the ribosome read the stop codon here. Then comes deletion. So if it is deleted also, again your frame is shifted. Instead of going like this 333, one is deleted, so the entire frame shifts. That also leads to entirely different protein. So these are the effects if the variation occurs inside a exonic region. If it occurs in the promoter region, then it is going to affect the way your gene is stimulated, because those chemical activators which will come and bind to the promoter will not bind properly. So eventually it will delay the transcription, it will delay the translation. Sometimes even it will stop the transcription and translation. So say for example here one particular position of the DNA is there, and in this position I had marked here as A. So in that position, this is the double stranded DNA is there. So A, it could be either A, G, C or T at that particular position. So how many different alleles could be there? Two alleles, right? Because maternal origin, paternal origin, you have two chromosomes. So in those two chromosomes, you have to see what is the allele present. So what are the different combination of genotypes that can be there? It can be homozygous for either of the alleles, or it could be heterozygous. This we have seen already. So we'll see how one of the applications it is useful. So we'll have a better understanding what is the importance of checking for SNPs. This is a particular variation. Okay, it is present in a gene called as CYP2C19 gene. Okay, what is this function of this gene means? This is an enzyme which is produced by the liver to break down a particular drug called as clopidogrel. Okay, so a patient who has undergone a heart bypass, and they have kept those tubes they'll keep, right, for preventing clotting. They keep those tubes and the blood flows through the tubes. So in that case, what happens? The doctor prescribes a clopidogrel for these patients, so that the blood, it will prevent the occurrence of clots. So the function of this drug is to prevent the occurrence of plaques or clots. That is the action. So once this drug is taken, the enzyme produced by the CYP2C19 goes and cuts or breaks this drug and makes it an active drug. After it becomes active, it will maintain the smooth flow of the blood, and it will not allow it to clot, because that clotting mechanism is there and it plays a role in controlling that clotting mechanism. So when doctors found that some of the individuals who were given this drug returned back to the clinic after some few months and claimed the same problem, and when they found that they had clot in that tube, but the patients have taken the drugs correctly, correct dosage, they did not miss anything. So this query led to the question of why some people are doing this and why some people are returning back. Well, whereas for others it is working very good. So they found out there is a single nucleotide change. Okay, if T is present, okay, it will lead to the formation of normal enzyme. So the patients who have TT at both the positions, they have no problem. The enzyme will produce properly, they will cut the clopidogrel drug properly, and the drug will act properly. But if a patient has C, even they have one C also, what happens? It will not function properly. What it will do, if it does not function properly, then clopidogrel will not become an active drug, and therefore even though they take the drug properly also, it will lead to the formation of plaque again. So this is a very simple test that could actually have an effect in the patients directly. Okay, so if, by just checking for the DNA of a particular individual, we can give them correct drug. So in that case, if they have C, we will not give clopi, we will give them some other drug. Okay, so it is very useful for the pharmacogenetics, the application of finding a SNP of a particular individual. Here comes the lactose intolerance. So there is a particular change in a gene, okay, LCT gene, which is responsible for producing the lactose enzyme, which will go and break your lactose. So if both the mother and father carry that particular mutation, and the other chromosome in them is normal, okay, so this is the mutation, this is the normal. So they are heterozygous at that position. Both of them are heterozygous, okay. So they will be lactose tolerant because one of them is normally functioning, the other only has the mutation. Whereas in children who get both the defective one, it will lead to improper production of the enzyme, and that will lead to lactose intolerance. And this is also because of the single nucleotide variation. Okay. Okay, we have some more time. Okay. Um, so how do we find this out? So how did we find that out? That variation in that CYP2C19, this is associated with this is associated with cases. How did we find that out? Because what we do is we define, how we do in research is we define the cases. So all those patients who returned back with the plaque, they form the case. And all those patients who are performing properly, they form the controls. So when we checked for the genes, so you know to a certain idea this is related to drugs, and usually drugs metabolizing enzymes are from the liver, and they know what all genes are responsible for that. So they search in those genes. So they amplify the genes and search in them. So they found out that here the representation of particular allele is more. So if you see in cases, A is more when compared to T. Here T is more. So this is how we tell that A is the variant associated with a case or a disease, whereas T is the allele that is associated with that of the control. So a person having T will be normal, whereas a person having A at a particular position will be more prone for getting the disease. So this is how we do research and we publish that data. Based upon this data, the company takes up those information and comes up with a particular panel for calculating the risk of the particular individual, which we'll see in the second session. So when we take all these things, genetic data and all these things, we have to make sure of one thing is some diseases are polygenic, some diseases are monogenic. As the name suggests, monogenic means, as I told you, cystic fibrosis, one single variation in a particular gene leads to the disorder or disease. Whereas in heart syndrome or the coronary artery disease, what we saw, there were multiple variations in different regions. So different genes are involved, but the effect of single SNP is less. Whereas in the monogenic disorder, the effect of the single SNP itself is more. That's why it is resulting in a phenotype. Okay. So in polygenic disorders, like in our nutrigenetics or the precision medicine, what we deal here is this case, the polygenic disorders. We check for multiple things and then we derive at a score which is called as polygenic risk score, and depending upon that we calculate and tell whether the patient is normal or is having a susceptibility for a particular disorder. So this picture was published in a Springer article where they have compared genetics with obesity, and you find here so many genes they found out. In that, if you see FTO, this is one important gene, and other MC4R one gene is there, BDNF one gene is there. The variations in this gene are very important. So what they do is they list down the variations. Okay. And then what they do is the variations which has the maximum effect, that is in multiple populations it is there, and in multiple studies have shown FTO gene, then that gene they give a particular weightage, that allele they give a weightage. Then the others. So in this diagram, if you see, there are several things, but only these highlighted ones are important. So they carry more weightage. And then what happens? We calculate the risk score. There is a particular algorithm. There are 2, 3, 4 different model algorithms are there. So the company chooses the best algorithm, feed this data. So a particular individual comes, we take their sample, we sequence all these genes, put them into that algorithm. The algorithm will calculate and tell whether they are susceptible or not. So this kind of studies are done by family-based studies, because we inherit our genes from our parents. So with that, we can find out, or the Jiva studies, like I told you, cases and controls they take, and they see which allele is present more common in the controls, which allele is more common in the cases. In that way also we can find out. So depending upon the effect size, so as I told you, single nucleotide variants, the effect size will be more, whereas our polygenic variants, SNPs, the effect size is very less, and multiple SNPs put together will cause the actual variation. And if you see that all these data that is available, about almost 80%, that is 78% is from the European population, that is the Caucasian population. And about only 10% of the published articles on these SNPs are from Asian population. So we, as a company, when we choose the SNPs, we have to make sure that such kind of studies have been done in our population also. Because an SNP, which is a risk in the European population, may not be a risk in our population. So you would have heard about how the humans spread, right? They originated from Africa, somewhere in south of Africa, and then the clade spread, one towards the north and one towards the south. The one who went north are the Caucasians or the Europeans, and the one who went towards the south are the Dravidians. So the Dravidians carry a different genetic makeup when compared to that of the Caucasians, because environmental factors are there, what type of food is available in that particular region, the climatic conditions, all these things slightly change that 0.4%. So we adapt accordingly. So allele which is called as risk in the Caucasian population may not always 100% be called as risk in our population. Sometimes it could be the opposite as well. So when we take such SNPs or choose such SNPs for our calculation, we have to make sure that we have sufficient studies from our population as well. And we should interpret that data accordingly. Okay. So there are different algorithms available for the calculation of the genetic risk score as such. You can go and see various algorithms are there. So we will not go into those algorithms. So this is how they score a particular individual, low risk, high risk, or medium risk, and then give the appropriate instructions. So all this genetic testing comes into the concept of precision medicine. So taking into all these components, you can give a better diet, personalized diet for your clients, by which your client can benefit as well. Okay, thank you. With this, we stop the first session. Ma'am, shall we take a break or we can move to the quiz?
Oh, we can go with the question. Am I audible?
Hello.
Oh, we can go with the quiz now. I request all the participants to be engaged in this quiz.
Hope the students have started joining. You must have used the software. You can just join. Then we'll go to the session as well.
Yes, ma'am. Yes, ma'am. They're joining.
So here the quick fingers is important. So each question has a few seconds time to do it. So once you read the question, click the correct answer and it will be scored accordingly. So let's take it on an easy note. So whatever we have heard so far for the past 45 minutes, we'll just put it to test. can we have 1 or 2 minutes break, ma'am?
Ma'am,
Ma'am?
am I audible?
Yes, ma'am. Yes, ma'am.
Students are actually attending the questions. So what are we supposed to do now, ma'am? Are we going to the next session or?
Yes, ma'am. We'll move on to the next session because we have crossed the limit. I think we have till 11. I have the next session.
Okay.
So I move on to the epigenetics session. If they want to take a short break, and then we'll continue with epigenetics session, ma'am.
Ma'am, we can proceed with the next session and then we can go for a break, ma'am.
Okay, ma'am. Sure,
Okay.
sure.
Thank you so much, ma'am.
Okay. So I believe you had slightly not a continuous session. So you were slightly busy in the quiz, and now we are all active and bright again. So we'll move on to a slightly different or the next level in the genetics. So we have learned what is the basics of genetics. So I hope my slide is in the slide share mode and it's moving as well, and I'm audible. So can somebody confirm that it is okay? I can proceed now.
Yes, ma'am, you're audible, ma'am, and slide is visible, ma'am.
Thank you. Thank you, ma'am. So we had seen the basics of genetics, how the DNA is, what is a gene and what is an allele, how it is arranged in the chromosome format, and how it is packed inside nicely inside the nucleus, and how it gets transcribed and then translated into the protein, right? So now moving on to epigenetics. So as the terminology suggests, it is something above genetics. Epi means above, and genetics means the basic DNA that we have read in the past 1 hour or so. So what led to the discovery of the concept of epigenetics? So, as in the quiz, you would have seen genetics along with environment plays a very important role, especially in the chronic disorders, right? So in epigenetics, what happened? When scientists were studying twins, basically they are monozygotic twins. That is, the same zygote got divided at the very early stage itself. So the copy of the chromosomes in both the zygotes were exactly the same. It was 100% the same. So monozygotic twins, you imagine they look very similar, But as they grow up, do you think they are still 100% the same, or they have different preferences? They are sometimes slightly tanned. One have a different preference for music. One has different preference for food, or etc. etc. One is a little susceptible for diseases, whereas the other is not. So the scientists were wondering how come they have that 100% they have the same DNA and same genes, and why are they having these small changes when they grow up? They don't have drastic changes, but small changes are observed. So that's when they led to the discovery of epigenetics. That is, there are some changes above the DNA, not in the DNA. Not there is no change. That is, they are not talking about any change in the DNA. That is something above the DNA that is controlling the way the DNA is being transcribed and translated. So that led to the concept of epigenetics. So in the embryonic stem cell, what happens? So that is your cells, embryonic cells, and you can imagine they are actively dividing. Okay. So first the ovum is there, the sperm fuses, and then you got the zygote, and it has to continuously divide. The DNA has to be replicated. Then it has to lead to the formation of, like, assigning a group of cells for forming different organs. Then it forms the brain, etc., etc., and then your baby develops as such. So in the initial phase, what happens? They are like runners. They just jump into action, all the genes. Okay. Then, but once they are differentiated, that is, after the cell has become designated as liver cell or hair cell or brain cell or skin cell, after that you can imagine not all the genes will be active. Only those genes which are required for maintaining this particular cell is active, whereas the others are not active. So that is called as switching on and switching off a particular gene. That is, when it is switched on, we call it as gene expression. The gene is expressing itself, and when it's switched off, it is silent. Okay, so you can imagine a brain cell is different from that of a skin cell. The skin cell, it produces melanin protein because it is required for protecting your skin against some harmful pathogens or anything. You know, it protects your skin. So that is the function of the skin cell. So that gene has to be active there. But that gene may not be active in your brain cell. It does not have any function there. So it is switched off. So, very early stage of development, all the genes are active, but as we move on, some are switched off permanently, some are switched on for some time and then active again. So that mechanism is controlled by something called as epigenetics. So this is one example wherein we have so far seen, no, we have getting two copies of chromosomes from each of our parents. So in this case, what happens? There is one gene called as IGF gene, which is responsible for the production of growth hormone. In this case, only one gene has to be active, whereas the other will be silenced. This is happening by nature. Okay. So it is programmed that one of the genes, it could be either one of the genes, it could be from the maternal origin or it could be from the paternal origin. One is silenced. Okay, only when that is happening, this has to happen normally, then the growth of the child is normal. That is normal fetal growth. When both are active by some mechanism, by some epigenetic mechanism, what happens? Both are active. So what happens? It leads to increased production of that particular hormone, which leads to increased fetal growth that will result in this syndrome, which is a diseased or a disorder kind. So epigenetic mechanisms play a very important role by how it is playing the game of keeping the normalcy inside a cell. It has to correctly switch on certain genes at the correct time. When the dosage is reached, it has to switch it off. That is how the cell is programmed, and that is how epigenetics works. This is a famous experiment on rats which proves that epigenetics is a concept which can be inherited, which can be modified. So in the DNA, if you see, there is a change in the DNA. As we saw in the previous lecture, cystic fibrosis was there. There is a change in the mutation in 7th chromosome. Okay. Going and editing the DNA is it is still in the trial stage. Okay, like that, the genetic technology has grown, but it is not applicable for all the disorders. We cannot go and actually change the DNA, and that leads to some sort of side effects as well. But much more easier is epigenetic modification. Epigenetic modification, if we can control that, to a certain extent, we can control the expression of that particular gene. And that was expressed in this experiment. Okay, so experiment one, you can see there are two sets of rats. One rat, the mother is very attentive, and the other rat, the mother is an inattentive. So the pups are very much stressed, whereas these pups are very happy. How they found out? They found out the expression of genes, the level of proteins in the hippocampus, that is the brain, and they found that the level of hormones were very high here, stress hormones, whereas here it was very less. The second experiment, what they did, they changed the environment. Okay, they interchanged the pups. Now, what happened? The phenotype reversed. That is, the stress levels came down for these pups, and here the stress levels went up higher. So, by changing the environment, they are changing the way the genes are expressed in the hippocampus, which is eventually leading to the change in the phenotype. So, as simple as changing the environment, you can change the way the genes are expressed, and that is the beauty of epigenetics. The third experiment, what they did not change the environment, but instead they gave a drug, okay, which is going to somewhat mimic the second experiment. And here, what happened? That epigenetic change was reversed, and here also the stress level came down for these pups. So by this, they proved that epigenetic changes are inherited. It could be changed depending upon the environment, like your interactions, medicine, diet, environment, exposure to toxic chemicals or disease exposure, exercise, microbiome, gut microbiome, all these changes have been proven to change the epigenetics of an individual, thereby changing the phenotype of the individual. So it could be sometimes beneficial, sometimes it could be harmful. So studies are done to identify those epigenetic changes which can lead to a beneficial effect and which can lead to a harmful effect. So this study was done, this was published in Nature, I believe. So right from the single fertilized egg to the death, that is old age and death, they had seen what all genes are active at different stages of the development. And you can see here, and they have mentioned all these genes. Okay, this is for a female they have done. And these genes were all active. Green means they're active, whereas red means they were silenced. So after certain stage during childhood, this particular gene was silenced. So there are different genes which are active at different stages, and depending upon that, the way we express our phenotype changes. That's why the twins have a different kind of phenotype at the later stage of their life. So what are these epigenetic changes? The well-studied epigenetic changes are: number one, histone modification; number two is the DNA methylation; and number three is the non-coding RNAs. Okay, so each mechanism we'll study. There are other mechanisms as well, and most of the studies well established is on histone modification, DNA methylation, and the more recent one is the non-coding RNAs. So what happens here is that, as I told you, it is epigenetics. That is, it is not change in the DNA, but something outside the DNA or on top of the DNA. So where the changes happen is that some chemicals are added to the histone molecules, and then depending upon that, it influences the cell expression. That is your transcription and translation. So these chemical molecules that are added by certain enzymes called as writers, and they are removed by something called as erasers, and they are read by some molecules called as readers. So you have some chemical moieties, say for example methylation, acetylation, etc., etc. So that is the chemical things that are added, read, and removed in a particular fashion, by these writers, erasers, and readers, and that's how it influences the DNA. So we'll see how that is done. First comes the histone modification. So here what happened, we saw that the chromosome is there. You can see these small bead-like structures. This is the electron microscope image. The DNA is there. These bead-like structures are there, histone molecules in which your DNA is bound. And then it forms the chromosome, right? So the histones, if you break up, they are nothing but having four parts, H2A, H2B, H3, and H4. These are the different histone moieties, and they form the core histone around which your DNA, that is about 147 base pairs of your DNA, is tightly bound. Okay. Then you have another histone H1 which runs inside where your DNA is joining. These histones have tails. Okay. These tails are the regions where your chemical structures, moieties like your acetylation and methylation happens. And that is what is causing the change in the way the DNA is expressed. So what happens here in acetylation? So acetyl molecule is added to the tail. In which position? It is added to the lysine residues. That is, it is a protein, right? So in that lysine residue, acetylation molecule is added by these enzymes called as HAT. That is histone acetyl transferase. So these are the writers. Okay. And the chemical moiety they are writing is acetyl group they are adding. Okay, and it is recognized by proteins called as bromodomains. Okay, these are the readers. So this will read the acetylation. So once the acetyl molecule comes and binds, what happens? These bromodomains read them, and then what they do is they reverse the charge of the histone. So I told you DNA is negative charge and histone is positively charged. So when acetyl group comes and binds, what happens? The charge of the histone gets neutralized. So what happens here? That tight bonding is not happening now. Since after neutralization, the charge is not that much, your DNA becomes loose at that particular point. It is not tightly wound around the histone. So that loosely bound DNA is called as the heterochromatic stage. Okay, it is in a relaxed stage. In that relaxed stage, what happens? It is made available for your signals to come and bind to the promoter region. Otherwise, it is very tightly packed and you cannot actually read it. So what happens when acetyl molecule is added by the HAT enzyme and then later read by the bromodomain, it will neutralize the charge of the histones and your DNA is available for the transcription and translation process. Okay. Then comes the methylation. This is the opposite of acetylation. So once the acetyl group is added, your DNA, that gene is ready for expression. It gets transcribed and translated and their protein is formed. So when enough protein is formed, the same example as ghrelin. So when your hormone, hunger hormone is produced enough and you have to stop, that is you have had food and satiety has been achieved, then ghrelin has to stop its production, right? So that is happening with the help of methylation. So acetyl group is removed. So erasers will come and remove that acetyl group. Then another set of writers will come and add the methyl group there. So what happens here? Once it adds the methyl group, that charge, whatever got neutralized is reversed and then again it tightly bonds. Okay. So this is the enzyme that is adding. This is the writer of the methyl group, that is histone methyl transferase. It transfers the methyl group to the histone tail and then it can be erased by demethylases. Here the readers, the acetyl group, it was bromodomain. Here it is chromodomain. You need not remember all these terminology, but and the concept happening behind. Okay, and why it is important. Okay, so the process of methylation, it leads to a much more tight, compact formation of the DNA, and the DNA is not available for transcription and translation. So the beautiful coordination between acetylation and methylation is what will switch on the gene and switch off the gene, and accordingly it will maintain the normalcy of a particular cell. And all these chemical molecules, that is the acetyl group and the methyl group, where do we get it from? We get it from the basic cycle that you have read in your nutrition. So breakdown of glucose to pyruvate that leads to acetyl-CoA, and this is the one which is going to give the acetyl group. And then somewhere down the lane, you will get the methyl group as well. Okay. So you have a set of writers, you have a set of readers, and you have a set of erasers. So by our research, we know which are the writers, which are the readers, and which are the erasers. And depending upon the particular given situation, so in this paper, they have talked about cancers and how the drugs, by altering either of these three moieties, they can control the cancer development or cancer growth or stop its growth. So we can easily modulate the expression of the genes. So here is the euchromatic stage where the chromosome is relaxed and you have lot of acetyl molecules here. And here you have the heterochromatic stage where your chromosome is tightly packed and it's not available. So you can see lot of methyl groups here. So acetylation means relaxed chromosome. Heterochromatic or methylation means a very tight chromosome and it is not expressive. It is not transcriptionally active. And there is another process called phosphorylation, which also acts similar to your acetylation. So this is with relation to your chromosome, and that we can find out by a technology called as chip sequencing. So by chip sequencing, we'll come to know which all the regions are acetylated, which all regions are methylated, and how it is associated with the disease. So this gives an overall picture. So here is the euchromatic stage where your acetyl group is added, and here is the heterochromatic stage where your methyl group is added to the histones. And you have your readers, writers, etc. Okay. So we'll move on. Next comes your methylation. Okay. In methylation, what happens? We saw all the changes that are happening inside the histones, right? So in histones, we had acetylation means transcriptionally active. That is the adding of acetyl group to the histone tails. Then if you add methyl group to the histone tails, it becomes transcriptionally inactive. So similarly, moving from histone modification, the second most importantly studied is the DNA methylation. So in DNA, you have ATGC, right? In that C, that is the cytosine, in that the fifth carbon, there you have this modification. That is the methyl group comes and attaches here in the fifth carbon of the cytosine molecule, leading to the formation of 5-methyl cytosine. So this is not changing the DNA. It is on top of the DNA. So it is still epigenetics only. How this is happening, like how you have writers, erasers, and readers there, here also you have the same. So you have this enzyme DNMT1, that is DNA methyl transferase 1. Then you have DNA methyl transferase 3A and 3B. So I told you epigenetic changes can be inherited. So that we are inheriting because of this enzyme T1. It will maintain the methylation status of the cytosine from the parent to the daughter cell as well. Meanwhile, we can develop new regions or new regions where C can get methylated. That is by 3A and 3B. Okay, that is de novo. So what happens? An important region in the DNA, you saw the promoter region, right? From the gene structure, that green color box. Usually before that, you will have lots of C's and G's, C's and G's. There will not be any A's or T's. Some 200 nucleotides to 500 nucleotides will be only C, G, C, G, C, G, like that. They are called as CPG islands. These CPG islands are present in front of a gene, usually at the 5' end of a gene. And those islands are very important to binding of the signals for transcription, etc. So when I told the DNA methylation occurs, the methyl group occurs and the C, it is natural to think that these C's will get methylated. But after studying and after doing this chip sequencing, they found out that CPG islands are generally not, prone for methylation. because they are very important for the gene to be transcribed and translated. Whereas other Cs are more prone for methylation. But in a particular case, we will see in the next slide where CpG islands are targeted specifically and all of the CpG islands are methylated. So here in the second picture, you can see this is a brain cell and this is a liver cell, how the methylation pattern differs between both of them. So depending upon these different methylation patterns, they express the different genes. So this was the condition I was talking about, which leads to specifically methylating the CpG islands. So you know that chromosome, females have two X chromosomes and male have one X and one Y chromosome, right? So if two chromosomes are there, they have the same set of genes, and it will be double the dosage for the female. So in order to avoid that, what happens by nature, here one of the X chromosome is purposely inactivated by the process of DNA methylation, wherein here all the CpG of the one of the X chromosome is heavily methylated. So what happens? It is becoming so heterochromatic. So you can see this is very different from this structure. So here everything is methylated and none of the genes here are active. Only the genes here will be active and they will produce the correct dosage of hormones. Whereas in the male, what happens? One X is there, one Y is there. They do need this one X for some function, right? So here, this methylation does not, or X chromosome inactivation does not occur. Okay. If it occurs, then it is dangerous. They are not getting the particular enzyme or hormone synthesized. So it will lead to some disorder. So this is one of the conditions by nature wherein the CpG islands are heavily methylated, leading to the inactivation of your X chromosomes. So next is, we have seen histone modification, we have seen DNA methylation. The third is the non-coding RNAs. So from the first session, you came to know about mRNA, which is the messenger RNA, which is nothing but copying the information from the DNA and bringing it out into the cytoplasm to form the protein or the enzyme. There are something called as non-coding RNAs. mRNA is a coding RNA. It will code for a particular amino acid, whereas non-coding RNAs means they don't code for an amino acid. When they come to the cytoplasm, the ribosome will not code it and give a rise to a protein or an enzyme. But why are they there? It is because sometimes what happens, transcription keeps on happening by some problem or by some mistake. So your mRNA keeps coming from the nucleus into the cytoplasm. But our cells are programmed in such a way that these non-coding RNAs, which are of different kinds, in that miRNA is the most important one, microRNAs. Okay, they are exactly complementary to your mRNA. Okay, so once your mRNA comes out into the cytoplasm, this miRNA will go and bind to it. They are complementary because A means here T will be there, G means T means instead of T, U is there, G means C will be there. Like that, they are exactly complementary. So once the excess mRNA is there in the cytoplasm, these miRNAs go and bind to it, and by a process, it will degrade or it will chop the mRNA. So even though transcription has happened, at the translational stage, our cells are programmed to control the amount of hormone or enzyme that is produced. So this is a very important mechanism where several miRNAs have been identified so far and have been synthesized also and given as treatment in cancers. So they will go and specifically bind to that mRNA and chop them, and thereby leading to stopping the cell division. Okay. So they are good therapeutic tools, these miRNAs. Along with that miRNAs, you have long coding RNAs, etc., etc. There is a list of non-coding RNAs, but the miRNAs are the important ones that have been well studied in this concept of controlling the expression of genes. So there are several factors which are involved, right from nutrition, paternal stress, that is during the first trimester of the baby formation, whatever we are exposed to, be it the smoke, dust pollutant, drugs, what we are taking, That's why we prescribe folic acid, right? For the females, they have to take it in the pre conceiving also, and after they have conceived during the first trimester, it's a must to take folate. And the environmental stress, all these will influence the epigenetic processes like DNA methylation, histone modification, chromatin remodeling, and non-coding RNA regulation, which in turn will act on the DNA as such, thereby leading to the change in the phenotype. Okay. So this is the same diagram that we have seen. So why are we talking about that in nutrigenetics? What is the importance of epigenetics in nutrigenetics? Because the acetyl molecule, the methyl molecule that is being used in epigenetics is sourced out of the food that we eat. So how did they prove this? So when they fed rats, pregnant rats with protein restricted diet, okay, they saw that certain genes showed decreased methylation. Okay, there was a certain set of genes in that particular pathway, and those genes showed decreased methylation. Decreased means what? The methylation is not that much. So what happened? The genes were active and they were able to produce. But when they, so the F1 generation for this particular rats, they have these decreased methylation, and the F2 generation also showed same decreased methylation, even though the F1 generation were fed the normal diet. So this is the parent rat which is pregnant. It is given protein restricted diet. The F1 generation, when they checked, they saw it had a decreased methylation. F2 also had decreased methylation in spite of F1 having a normal diet. So this shows that what influences our grandparents had, that is passed on from generation to generation, and it is being transmitted to our granddaughters as well. So by controlling all these environmental factors and exposures, we have to imagine whatever changes we are undergoing, that is going to be carried forward to the following 2 to 3 generations. The next example was in primates, where they gave high fat diet to the primates. Okay, that led to fatty liver in the first generation because there was increased acetylation at this histone. So we saw different histone molecules, right? So this particular histone was having increased acetylation. So what happened? There was reduced the production of HDAC-C1, which led to fatty liver in the fetuses. And obesogenic high fat diets explained that experimental animals are also associated with increased methylation. So all these kind of different diets given to the primates, they found out that changed the way from a normal primate to another primate which has a high fat diet in. And what was the phenotype because of that? And that led to a fatty liver. And why this is important? The SAM requires folates like B12, methionine, and choline, and that is an important donor for all these chemical molecules which are causing the change in the epigenetics. So these are the different components. You can go back and check various papers. They describe the various components in which food they are present, and what will be their effect on the erasers, writers, and the readers. So HAT is histone acetyl transferases, histone deacetylases, that is the erasers. So what will be their effect on it? So when we know that this effect, accordingly we will modify the diet of the particular individual, thereby controlling the epigenetics of that individual. And we can reverse it as well. So these kind of modifications led to a obese individual, but by controlling them, we can reverse the phenotype to a normal phenotype as well. So that is the beauty of epigenetics. So leaving rats and primates, how did they prove that in humans? So the first evidence in humans was this study of Dutch famine. I believe you will have it in one of your, during your course as well, you would have read about it. So 1944 to 1945, there was a severe famine, which and they found out that children born to mothers exposed to that famine, had a very high risk of certain chronic diseases at a later stage when the famine was over. But those mothers who were pregnant during this stage, 1944 to 1945, their children showed altered expression. What were the altered expression? The insulin-like growth factor showed less methylation, increased methylation of these two genes, and decreased methylation. So that, like that, they have several things. But when they compared children born to mothers exposed to Dutch famine and those born to normal mothers who were not exposed to this famine, they found out that they were highly at risk for coronary artery disease and obesity when compared to the children born to mothers who were not exposed to the famine. So that's how environment and diet also plays a role in changing the way the genes are expressed, and this could be easily modified. I would not say easily because studies are still going on, but have shown results by controlling the environment and by controlling or by giving certain drugs or by changing the diet, we could influence the way epigenetic modifications are happening, and they keep happening throughout the lifespan of an individual, right from the fertilization to the death. There are several stages at which epigenetic modifications are happening, and depending upon the correct exposure, we can keep on certain genes and switch it off at the correct point. If that switching off does not occur is also a problem. Switching on does not occur at the right time also is a problem. So the beautiful symphony by the epigenetic mechanisms is what is controlling the normal development of a person and normal healthy living of an individual. So with this, we have come to the end of the second session. And ma'am, shall we proceed to the quiz? It is about 11:10 right now. I can just take few questions, not the entire thing, so they can have a better understanding. Or we can have a clarification session. If they want to ask any questions, they can type in the question chat box. I can answer that as well. Hello. You can hear me, ma'am? Yes, ma'am.
Ma'am, we can go for a small session of quiz now for 5 minutes.
Oh.
Then we can have a break. Then we can proceed with the other session, ma'am.
Okay, sure, sure.
Okay. Or else,
I will ask.
we can proceed with an interactive session. If students having any doubt, they can ask you. So
Because it
till
is very
5
short
minutes, we
time.
can do that.
Yes, ma'am. Just 5 minutes is there. If we go to quiz, it will take a much longer time. So
Okay.
if they have any questions, they can put it in the chat box or they can ask me, ma'am. I'll try and answer them.
Yes, ma'am. You can give us the already students are having the QR code. So during
Okay.
the break or after the session, let them complete the quiz. We'll make sure that they will complete the quiz, ma'am.
Okay, ma'am. It was just
Okay.
for a jolly purpose that they have boring session they are hearing, no. So I want to be make them active. That's why we put up the quiz session. So if they want to continue, it's okay, ma'am, because the other person is actually sharing her screen. From my system, I could not do it. I'll ask her to just share the screen, ma'am.
Yes, ma'am. We can go for an question and answer session. If anybody have any questions, girls, if you have any questions, please you can ask, ma'am, or else we can go in for a break. Okay, I think there are no questions. We can start with the quiz then, ma'am.
Yes, ma'am. Yes, ma'am. Just some 10 questions I'll finish in the quiz.
Yes.
By that, the time will be correct, and then we can go on for the break, ma'am.
Yes, ma'am. Yes, ma'am.
Thank you, ma'am. Thank you. So
Well, please
we'll
start
start.
the quiz.
Yes, we can again join the quiz. This is on the epigenetics basically. So, what enzyme is responsible for copying the methylation marks from the paternal to the daughter strand of DNA during replication? That is, which is enzyme is responsible for copying it exactly, maintaining the methylation? Great. So 57% have given it as DNMT1. Yes, you're right. 3A and 3B are required for de novo. Okay. Moving on to the next question. Great. Congratulations once again. What is the primary role of epigenetic marks in the context of gene expression? Yeah, next, please. Yes, great. So everybody has listened. I'm very happy. Regulation of gene activity without altering the DNA sequence. That is the right answer. This is on top of the DNA. Very much. Thank you very much. The next question. Great. Congrats. What enzyme is responsible for copying methylation marks? I think it's the same question once again. DNMT1. So the percentage has improved now, 87%. Good. Next question. Congrats. What is the association between obesity and epigenetic age? What do you think is the relationship? Great. Epigenetic age is higher among people with obesity. You're absolutely right. Next question. Congrats. Which of the following diet-derived components have epigenetic effects? Exclusively, we have not told, but it was on the slides. Good. Very good. All of the above. Yes. Next question. What best describes epigenetics? Absolutely right. Study of heritable phenotype changes not involving changes in the DNA sequence. Yes. The next question. just few more we will take. What is the primary role of epigenetic markers in the context of gene expression? I think this is also a repeat question. Can we move on to the next question? Yes, yes, yes. Very good. Next. Which of the following best explains why identical twins may develop different diseases later in life despite having the same DNA? So epigenetic modifications is explaining why some of, even though the identical twins are having 100% same DNA, they have different phenotypes at a later stage in life. Yes. The next question. Can we move on to the next question? Okay, I think there is some network issue. Ma'am, we'll wind up with this. Thank you once again for the wonderful opportunity, ma'am. And I thank all the students for their active participation in the quiz. And I'm happy to see that you have got the concepts correctly. Request, ma'am, any doubt, please forward the questions to me and I will answer them as quick as possible. Thank you, ma'am.
Thank you so much, ma'am. Your words of wisdom have inspired us. Also, you have added immense value to our gathering about basic instruction about the gene, then the indispensable role of genetics in medicine, and how to use it in precision nutrition, and about the epigenetics, and so on. Thank you so much, ma'am, for your valuable insights. Thank you, ma'am.
Thank you.
Thank you so much for joining, ma'am.
Thank you once again, ma'am.
Yes, thank you, ma'am.
Greetings to one and all gathered here. I'm Miss Roshni from First MSc FSMD. It's my privilege to welcome you all on behalf of the Department of Home Science to International Online Workshop on Decoding the Science of Nutrigenetics, Session 2, Clinical Applications of Genetics. Education is the most powerful weapon which you can use to change the world, said Nelson Mandela. Let's move on to the next speaker of the day. I request Catherine Subhashini of First MSc FSND to introduce the second speaker of the day, Dr. Janani Tamilvarnan ma'am, to the audience. Good morning, all. Dr. Janani Tamilvanan is a renowned nutrition expert and director of the Institute of Nutrigenetics, a leading education forum promoting research and education in nutrigenetics. With a strong educational background, including a PhD in food science and nutrition from Avinashilingam University, India, Dr. Janani has established herself as a leading authority in the field of nutrigenetics and personalized nutrition. With over a decade of experience in research, education, and industry, Dr. Janani has trained and certified over 3,000 dietitians and reached out over 1 lakh students through Reach Out program. Her expertise lies in translating scientific evidence into personalized lifestyle recommendation for health promotion and disease prevention. Dr. Janani's research focus is on nutrient-gene interaction in nutrigenetics and nutrigenomics. She has published several papers in reputable journals and has received numerous awards for her work, including the Best Presentation Award at the International Conference on Nutritional and Genetic Epidemiology. As a pioneer in the nutrition field, Dr. Janani aims to make personalized nutrition accessible to everyone, and her work has been recognized through partnership with leading institutions and organizations. Thank you. Thank you, Catherine Subhashini. Now I request Dr. Janani Tamilvanan ma'am to take over the session.
Hi, good afternoon, everyone. Hope I'm audible. Am I audible? Can I continue?
Yes, ma'am, you're audible, ma'am.
All right. So, good afternoon, everyone. And it's a pleasure to be here today on the International Online Workshop on Decoding Nutrigenetics. And I thank Dr. Vijay Bahani ma'am for this opportunity, and to all the students who are actually part of this workshop today for actively participating. And we look forward for you to learn something on nutrigenetics when you leave the hall today, and to pursue something if it interests you. So this is a small initiative to spark interest and for you to show the path to explore further if the science of nutrigenetic interests you. So without further delays, I would just start my presentation. So it will be more interactive. I'll just present. Is it visible?
screen is visible now.
Yeah. So today it's just a short discussion about what nutrigenetics is all about. Dr. Sharmila would have explained you about the very basics of genetics to start with, about the terminologies. Now going on to the science of nutrigenetics. So as nutrition students, it is very important to know about this emerging science of nutritional genomics, which is kind of picking up in India very well in par with other countries in the world. So as young minds, if you are looking into the molecular nutrition aspect, it would be helpful for you to understand in depth about nutritional sciences and how it is kind of intertwined with the science of genetics. So the future of personalized nutrition, so this is how we think it's the way forward. We have been stratifying or tailoring diets based on group of individuals based on population-based dietary guidelines. So we slowly moved on to something referred to as personalized nutrition, where we also take into consideration about individuals at an individual level. So we believed that individuals, if they are looked at as a unique identity, then it would help the health professionals as well as the individual to get advice based on their individual body type or genetic makeup or gut microbiome. But the way forward or the future is what we are expecting, which would involve sufficient understanding about the complex interrelationship between microbiome, genetics, transcriptomics, which is the genotype of the person, as well as the phenotype, which the assessment is in the current nutritional assessment or the traditional methods have been following, which is based on the symptoms. So if you look at the transition that has happened from stratified or tailored nutrition to personalized nutrition, so it has been very transformative, where we thought it would be more effective in delivering nutritional plans rather than basing it on population-based dietary guidelines. And it is also believed that due to genetic advancements and genomic sciences, we believe that precision nutrition approach is something that would be more precise in nature, more effective, and it will be also kind of a strategic plan to deliver public health care as well. So this is the difference between one size fits all versus personalized nutrition approach. So if you look at the one size fits all, we have similar population-based dietary guidelines, which is issued by authorities in different countries, which assumes that the individual requires similar amounts of food from different food groups, and this we believe that it would be appropriate for everyone. Whereas if you look at the personalized nutrition approach, you're looking at an individual at different layers or different aspects of the individual, like the genetics of a person, like the gut microbiome of the person, or the metabolomic nature of the person. So we do the profiling and understand at an individual level about the requirement of the person from different aspects. So this is the difference between one size fits all versus personalized nutrition approach, where the individual nutrition is given more importance than assumption that a kind of population-based dietary requirement would be sufficient enough. So the rationale behind personalized nutrition approach is very simple. We are trying to answer the question, why do individuals respond to the diet in a different manner? So why this variation comes in, or why there is a difference between individuals? So why a person is able to live longer and why a person is not able to live longer? It's also about the health and longevity issues. So what determines the lifespan of the person? So we are trying to answer many questions using the personalized nutrition approach. Though I want to pinpoint here, personalized nutrition could be used in different contexts, but here in today's session, we are confining ourselves to personalized nutrition based on the genetic makeup. So when we look at the conventional meal plates, so I'm very sure that you're all dietetic students, so from different levels like first year, second year. So most of you would know about the ABCDs of nutrition. So when we look at the traditional approach, nutrition approach, so we look at the anthropometry, biomedical characteristics, clinical signs and symptoms, and dietary assessment to assess a person's nutritional status. So based on these are the parameters which goes inside the meal plate of the person. But when we look at personalized meal plate based on genes, so these are the aspects which are included and which are integrated as part of the meal plate. So which means that, in addition to ABCDs, you're also integrating genetics into your meal plate from now on. So that is the main difference. It's not that genetics can be done in isolation, so it doesn't perform any function in isolation. The phenotypic assessment is what the current nutritional assessment is all about. So we assess the nutritional status based on whatever is descriptive, whatever is measured. So whatever is seen outside, but what was not there is the genetics part, which we are integrating because of it is available now. So we are able to test it, we are able to analyze the genetic information, and that is why we are going to integrate along with the current ABCDs of nutritional assessment. So this actually refers to the genetic variability among humans. So we are all the same. We are all referred to as humans. But how are we different? It's only 0.1% difference between us, which makes us really unique. That is human metabolic individuality. So that is actually contributed by the genetic variation that is present in the humans, and that is what comes as the baseline for nutrigenetics. So just because people are different based on the genetic makeup, and this genetic variability is contributing to nutritional genomics. And here I would also like to differentiate between the two terms, nutrigenetics and nutrigenomics. Though these two comes under a bigger umbrella called nutritional genomics, these are like two sides of a coin. So you can't actually separate them. But people are using it interchangeably, nutrigenetics and nutrigenomics. And here in this session, try to learn about the difference between nutrigenetics and nutrigenomics. So if food is affecting gene expression, then it is nutrigenomics. I'll also tell you about certain examples for you to understand about nutrigenomics. Whereas the genetic variation, I told you now we are all different in terms of genetic makeup, that is 0.1%. And SNPs, as actually described by Sharmila in previous session, so you know what are SNPs now, single nucleotide polymorphism. So the SNPs, they actually contribute to the way we respond to food. So we are all different when it comes to food. So you might metabolize carbohydrates differently, or the other person might metabolize it differently. So this different is attributed by the SNPs present in our genes, and these SNPs contribute to the difference in the response. So these SNPs, how it determines the response to the food that you consume is referred to as nutrigenetics. Though it might sound similar, I'll just give you an example. So we don't just stop with how food interacts with genes, but we are also more focused on how these gene-nutrient interaction is having an impact on diseases like cardiovascular or diabetes or obesity or any non-communicable or any kind of diseases. So that is the reason why it is more a personalized nutrition approach and a preventive nutrition program where we are looking into more of prevention rather than managing the condition. So to understand it better, I'll give you an example of nutrigenomics. So I said food, when it affects gene expression or when it affects the gene activity, it is called as nutrigenomics. So you can actually look into vitamin C. So it actually regulates the gene encoding for the extracellular matrix, which actually helps in the lipid oxidation. So that is why we keep talking about antioxidant and then to consume vitamin C rich food to improve immunity. So it affects the food here, the nutrient here is vitamin C, and it regulates the expression of genes affecting the gene expression. So likewise, there are few nutrients that are mentioned here, you could see. So for example, vitamin B12. So it helps in the methylation of DNA. And if you look at iron, so it's a part of the ribonucleotide reductase, so various enzymes that are involved in the mitochondrial processes. So, and zinc, it helps in RNA synthesis. So these are few examples. So now we are talking about how one particular nutrient will affect gene expression. So this is just one part of the genomics. So it's just, we are talking about DNA. But food acts as a signal. So at a cellular level, it needs to get absorbed. And for that to get absorbed, you need transcription factors where it takes the nutrients and then it pushes inside the cell to get utilized. So when you talk about the transcription factors, the collective, all the mRNAs together. So these help in the transport of the nutrients and the nutrients from the cell to the, it helps the cell to utilize the nutrients. So in case if you're learning about how nutrients are affecting the transcriptome or transcriptome, how it influences the response to particular nutrient, it's called as nutritranscriptomics, which I've mentioned here. And the collection of proteins from the cell, it gets utilized, and from the mRNA, it forms protein. And the collection of protein, a complete set of protein which are expressed, is referred to as proteome. And once you learn about how nutrients affects the proteome, it's called as proteomics. And then it's like all the metabolites which are formed during the cellular processes as a result of metabolism, it's called as metabolome. Once you learn about how nutrients are affecting these metabolomes, then it is referred to as nutrimetabolomics. And I hope you also learned about the epigenome in the previous session. So that will also be kind of how chemical modifications happen in the DNA and how it changes the gene expression. So all these are coming under nutrigenomics, whether it is DNA or transcriptome or proteome or metabolome or epigenome. So it is all termed or it is referred as nutrigenomics. So this is like a relationship where we are studying how the food that is consumed is affecting all these regions of the genome. But you are now focused on the other side of the coin, which I mentioned as nutrigenetics in today's session. I would like to take one example for you to differentiate, to understand the difference between nutrigenetics and nutrigenomics, with folate as an example. So do you remember where folate is highlighted usually? So what is basically folate? Can I have some chat messages or someone can tell me? What is folate basically? I don't see chat messages here. Anyone?
It's vitamin B9.
Yes, it's vitamin B9. So what happens is when you consume greens or any kind of leafy vegetables or meat liver or any kind of pulses or legumes like soyabeans, rajma or chickpeas, so any of the legumes, so you have folate in it and it gets converted. I'm not a biochemistry professor, but I'm just like, I'll put it in very simple terms so that you understand the difference. Because whenever you speak, you say nutrigenomics, sometimes nutrigenetics, but there is a lot of distinct difference between the two terms, and I want you to learn about it and use it appropriately. So when you consume these greens, so once it goes inside the body, it gets converted to tetrahydrofolate, and further it gets metabolized to 5,10-methylene tetrahydrofolate. So this is a precursor for thymine molecule. I hope you remember about thymine, right? So she would have taken the DNA structure, Dr. Sharmila would have highlighted about the DNA structure. So this is very important for DNA synthesis because it's a precursor for one of the DNA nucleotide. So if you don't eat greens, especially during pregnancy and lactation, we highlight about the consumption, because pregnancy is a time where there is synthesis happens at a very rapid rate in the woman's side. And that is the reason why we make sure that folate is supplemented during pregnancy. Meat, liver, or greens are actually emphasized so that the DNA synthesis is actually proper during infants or during the pregnancy stage. So here you're looking at how a particular nutrient, in this case it's a folate, and how it is affecting DNA function. Whereas on the other side, the same 5,10-methylene tetrahydrofolate, which acts as a precursor, is also responsible for the production of 5-methylfolate, which requires an enzyme called as MTHFR, methylene tetrahydrofolate reductase. And you know that enzyme is made up of proteins. Once it's made up of proteins, it is actually given instruction by a gene called as MTHFR gene. So this MTHFR gene produces, encodes an enzyme, which is MTHFR enzyme, and that is responsible for the conversion of 5,10-methylene tetrahydrofolate to 5-methylfolate. And this 5-methylfolate is important for the conversion of homocysteine to methionine. So why we are talking about homocysteine to methionine? Because homocysteine has to be kind of in a kind of homeostasis for good heart health. So in case if you have a variation, there are 3 types of variation a person can have. So if you have the first type, you might have normal conversion efficiency. Second type, only partial. And third type, you don't convert it at all. So in this case, if you are converting it properly, then you might require only normal amounts of folate. But if you belong to the second type, you would only convert half of it, which means that you might be deficient and your homocysteine levels can go up because you won't produce adequate amount of 5-methylfolate. And third type is you don't convert it at all. They are at a very high risk of having elevated homocysteine levels. So here, what we are doing is, for the first type who has normal conversion efficiency, normal amounts of folate as recommended per day could be given. For the second type, where they don't convert it or they convert it partially, we can directly supplement them with 5-methylfolate, because 5-methylfolate supplementation would help them to surpass this conversion. And you can also tell them about it is necessary for the maintaining the normal levels of homocysteine levels. So this is another thing. And third one, who are at high risk, can be given 5-methylfolate in addition to methionine restriction, because methionine is an amino acid which has the tendency to recycle back to homocysteine. So here, not everyone requires the same amount of folate, because every pregnant woman in India is almost given the same dosage of folate supplement, which is not so, it's so right, because everyone would have a different requirement. So the genetic testing is helping you to identify which group you belong to, and thereby you can give the supplementation accordingly. So it helps in dosage differentiation. So the first group is going to get the normal amounts of folate, then you can suggest them with dietary sources. The second one, a different form of folate. And the third one, you're altering the recommendations. So you can add vitamin B complex as cofactors in folate metabolism. So you can also add those food sources. Betaine and choline also provide a kind of, it will help in folate metabolism. So that could also be supplemented. So these are something that you are learning about molecular nutrition. So here your genes actually determine your folate requirement. So this is nutrigenetics, whereas this side, food affecting gene, is nutrigenomics. Hope I'm clear about the difference between nutrigenomics and nutrigenetics. So from now on, when you're talking about SNPs and how it influences the food you eat, that refers to nutrigenetics. Whereas when you talk about how food is influencing your gene expression, it is termed as nutrigenomics. So nutrigenetics is nothing but you are understanding the human metabolic individuality by understanding a little bit of molecular nutrition or biochemical pathways. So if you look at folate, as I said, this is folate cycle, but no cycle is linear in nature within our body. It's so complex because it is also linked with other pathways. So if you look at this folate cycle, here you look at vitamin B12, and when homocysteine is getting converted to cystathione, which is another pathway, you require vitamin B6. So if you understand, instead of only supplementing a woman with folate during pregnancy, by understanding her genetic variation, we can also add these recommendations in the diet planning. So supplementation with 5-methylfolate, methionine restriction, vitamin B12 and B6 rich foods, because these are cofactors for these enzymes which are involved in folate metabolism, and betaine and cysteine rich foods. So there's also another pathway which gets into the homocysteine pathway, which is the betaine and cysteine. So then we can also suggest these recommendations if they want to improve their folate levels in their body. So I hope like this was given by Sharmila. Now you would have understood what is a genetic variation, but a simple analogy. If you look at an organ system within a cell, if you look at the nucleus, and within the nucleus, you will have the chromosome. And when you untwine it, you will have the DNA. And within the DNA, you have these. So what we are looking at as an analogy is imagine a genome to be book. And you have 24 chapters, those are chromosomes. And you have pages which makes up those chapters, which are the DNA. And words are the genes, letters are the nucleotides. And spelling mistakes are the genetic variation or the SNPs that we are going to talk about. So imagine like there are two individuals amongst you. That is, one is having A here, adenine, and the other individual is having guanine here. So if one is replaced with another nucleotide, an individual, this is what we are referring to as a single nucleotide polymorphism. And this is what is contributing to the difference in how you respond to certain food. So it's not necessary one has to be replaced with always another nucleotide. One nucleotide can be replaced with many, or one can be replaced with none. But all these are referred to as single nucleotide polymorphism for it to be present in more than 1% of the population. But if it is lesser than 1%, it is termed as a rare mutation. So that is totally different. Here we are talking about single nucleotide polymorphism or single nucleotide variations. Those are called as the SNPs. So now you would have understand. So far, whatever assessment we have done in nutritional status assessment or dietary assessment, it would be kind of involving the dietary intake, physical activity, various environmental factors like affordability, culture, preferences, and all that. So those are referred to as phenotypes. So whatever measurable characteristics that are descriptive, that are seen, that can be measured is referred to as phenotype. And we have been doing all phenotypic assessment because it was only available. And now it is because all these genetic testing gene information of a person is available, we are trying to integrate it. So this is the way forward, as I told you, precision diagnosis. But in today's session, we are looking at a very tiny part of genomics, which is the nutrigenetics. But you have so much more to it. I'm just giving you a holistic view so that you could understand the depth of the science and how it can be integrated into nutrition. Nutrition will become the core element of healthcare in the future because this is what people are going to rely on. So food is the ultimate medicine, and how we are going to integrate all these omics approaches into food is what brings in the complexity and the challenge. So we are looking at, there is so much more to look into a particular individual, and that is why we call it as precision diagnosis, where you cater to individual nutrition treatment plans for nutrition. So when I spoke to you about nutrigenetics, I should also tell you genes doesn't function in isolation. Genetics is not a destiny for anything. So just because you're born with certain genes doesn't make you susceptible to a particular disease. It is not that you will get the disease. So it is not a destiny. You could still modify, you could still play around with, because there's a constant interaction between genes and environment. I'll try to explain with this health pendulum. So where you imagine this is a health pendulum, on the left you would find certain factors that are not so good, harmful factors, maybe we can call it. So genes which makes you susceptible to certain diseases like diabetes or obesity, and poor nutrition, food components like the way you eat food, food patterns, or bad lifestyle. On the other side, you have good factors like protective SNPs, protective genes, good nutrition, you take in good food, and there is good activity, physical activity every day. So when you have more of the red ones, it pushes the pendulums towards the disease. When you have more of the green ones, it pushes the pendulum towards health. So this is explaining it, and majority of us will have combination of both red and the green ones, which makes us in the middle. So if you want to go towards health, take more of these factors into your lifestyle. And if you want more towards your disease, then you can choose. So it's about the choice. So it's not about the genetics, it's not about the lifestyle, it's more about the choice, because everyone, it's a combination of both your mother and the father's genetic material. So you might have certain good genes, you might have certain susceptible genes, but it's also about the choices that we make, which pushes our health towards us, towards health or towards disease. So here, when I talk about nutrigenetics, I should also tell you that the evolution of humans in the food system. So we need to understand a little bit. So genes didn't change over time. It also got adapted to the external environment. So when you look at hunter-gatherers, they used to run for food. They had larger canines, sharper canines, like teeth used to come out, and they used to run, and they were more muscular. They were more like the body built was like the frame was larger, because they ate what was there, the whatever food was available. They didn't eat all 3 meals every day. So whenever there was food, they consumed it, and it was a kind of an intermittent fasting. So you could see that whenever there was food, they consumed, otherwise they used to be on hunger pangs. So it is like a cycle of both fasting and feasting. And slowly they started to settle in places of civilization where they had rich water sources, and humans began to grow crops. And after the discovery of fire, where cooking happened, so processing started to happen. So you could see that it was more of unprocessed natural food when they were hunter gatherers. And when humans became agricultural pastoralists, so it was minimally started to get processed after the discovery of fire. And now today it is mostly Swiggy and Zomato, which is giving you food on the table. You don't even have to move your part. So it means that the energy expenditure for acquiring food has drastically reduced. And also you could see that the processing has certainly increased. So if you see the food that you consume, it's totally 100% or 200% processed. You don't get raw food at all unless you have a home garden, and also the soil, everything is totally contaminated, and it is like fully processed food is what. And the cooking as well as the way we eat also has changed. So hunter gatherers used to bite, chew food, where they increase their saliva secretion whenever they ate food. But today we just gulp food, dosa, idli, bread, pizzas, or chicken. So everything is totally processed. So we need to also accept that there are a lot of changes that happened in the environment that accompanied the that accompanied the changes and made our genes to get adapted to the external environment. So if you also look at the food availability and its impact on our genetic patterns, so you should understand that it is also about the origin of humans, that all humans originated from Africa, and then they took different routes of migration. And during this travel and migration happened, so you could also see that the different food that was available in different regions, that played an important role in the availability as well as in the genetic difference in the genetic patterns that existed today. So it's about weather conditions, it's about the food availability, it was also about how they consumed it and how it was available. So if you look at these could have contributed to the genetic differences in different parts of the world, and that is why we can see that the differences in the genetics is actually influenced by the environment. So why ethnicity is playing an important role when it comes to genetics is that because of all these differences and what your ancestors ate would have definitely has a kind of an influence on what you are eating now, and also like how is your genetic makeup. So this actually is an important indicator to understand the concept of gene nutrient interaction. So it is always the nature, that is the genes that you're born with, and nurture, that is the environment you get nurtured in. And that certainly has an impact on your genetics. So it's not about just the genes, it gets transferred from generation to another. It constantly interacts with the environment, and that has an impact on your health and the genetics. So nutrigenetics is not something new. It has been there for decades together. You could see that gene nutrient interaction in phenylketonuria. So, I mean, we have been treating this condition PKU ever since nutrition has evolved. And the simple treatment for this is to restrict high phenylalanine in foods and to opt for low phenylalanine foods. So this is like you experience PKU with people having a defective PAH enzyme. So where the person's blood has elevated levels of phenylalanine, and that is leading to certain symptoms in them, like it could be related to their cognitive skills, or it could be like gastrointestinal symptoms. So once you restrict the phenylalanine, these symptoms get reversed. So this clearly shows that so it is all about genes and food constantly interact, and the way that we are looking at it is so once you make changes in the food, it's very obvious that it can be actually it can be rectified. So this is with particular to one gene that is called as PAH gene, or phenylalanine hydrolase enzyme, which is encoded again by the PAH gene. So here we are looking at one particular gene that is responsible for encoding and one gene which is responsible for this condition. So once you make the rectification or modification in the diet, this gets reversed. But the complexity arises only when there are multiple genes with multifactorial factors, multifactorial conditions like obesity or diabetes or hypertension, where you have like a lot of factors which are contributing to it, and also a lot of genes which are contributing. So this is called as a monogenic condition where there is a single gene involved, whereas when we look at the other condition, there are multiple genes and multifactorial in nature. So this is what I mentioned about multiple genes. So most of the population in India, they are vitamin D deficient, and we keep talking about vitamin D deficiency because we have started to spend more of our time indoors rather than getting sunlight exposure. So apart from a nutrient deficit for vitamin D deficiency, we also see that we don't get adequate sunlight today. That is the reason why there is a kind of increasing statistics which shows that there are many more Indians, only 75 to 80% of the Indians are vitamin D deficient. And if you look at the metabolism of vitamin D, either you get from food source in the form of ergocalciferol or from the sun in the form of holocalciferol. This is termed as vitamin D2, and this we call it as vitamin D3. So once it reaches the liver, it gets hydrolyzed into calcidiol, and then in the kidneys it gets further catabolized, and then either you get calcitriol, which is the primary active form, and then it goes into the circulation, and with the vitamin D receptor it gets utilized, and it goes into the cell. I mean, it is utilized by the cell. So this is a process in short. So if you look at the genes that are involved in vitamin D metabolism, so you could find almost more than 10 genes that are involved at each step. So for the hydrolysis to happen, or from the time it enters your body to the hydrolysis process to further reduce to calcitriol, you could see like these are the genes that are involved. So it's not like PKU where it's only one gene, but you could see there are many genes involved. So any defect in any gene could cause vitamin D deficiency. So it is about finding the root cause for the deficiency and then addressing it. Then it would become more appropriate and effective in nature, because once you get the genetic test analysis, so you would be able to find whether it is the DHC reductase or VDR, or what is the reason behind vitamin D deficiency. So it will be easy for a dietitian to address and to solve the deficiency status of an individual by identifying which gene is actually defective in nature and which gene could be rectified. So the next question to you as a nutrition student is, mostly when we look at, I'm going to give you some examples to understand this nutrient-gene interaction. So often the question would be about which oil is the best, whether it is coconut, gingerly, or whether it is olive oil or walnut oil or any kind of oil. But in the past, we've been hearing to this question. It's not only for oils, it's for fats, it's for any type of fat like egg yolk, whether I have to have egg yolk or just egg white. So these are some dietary dilemma which has existed for centuries now. And now, because of the personalized nutrition approach, the question has changed to which oil is good for me, because it makes a lot of difference. So to understand this, whatever the plant source based oil that you're consuming, whether it's going to be peanut or whether it's going to be gingerly or sesame or olive, so it has to get converted to EPA and AA in your body to get utilized by the body. And that happens with the help of series of enzymes like FADS1, which is fatty acid desaturase enzymes. So these enzymes, it's like one is there and then two, and there is another gene which is involved, ELOV2. So these are genes which are encoding these enzymes which are involved in conversion of short chain to long chain fatty acids. So any plant-based oil that you take in, it will have short chain fatty acids which has to be converted to long chain fatty acids to form eicosanoids, which are good. So you could actually find EPA, DHA in all the infant foods, because EPA and DHA is actually responsible for the cognitive development. So it is always highlighted in infant foods. And body does it every day when we consume food if it has fats in it, because this has to happen on a regular basis. Every cell needs this, and this is happening. So if you have a genetic variation, if you carry A allele, those people are good at converting these short chain fatty acids into long chain. But when you carry C allele, those people are not good in converting the plant-based sources to animal-based sources. So by genetic analysis, you will be able to differentiate who is carrying A allele and who is carrying C allele, and accordingly, you can give recommendation. So if a person is carrying A allele, they are likely to benefit with plant-based sources, because they'll be good in conversion of short chain to long chain fatty acid. Whereas if they are bad or poor in conversion, they can go for animal sources, because animal sources readily contain EPA and DHA in their diet. So what happens here is we are trying to distinguish or stratify the individuals in the population based on the genetic makeup, and then suggest dietary recommendations based on that. So next one is, so I would like to conclude that, so if you are relying on genotype-based recommendation, it is easier rather than biasing our recommendation. So there are 200 plus people, students here. So each one of you will have their own thoughts on which oil is good. But rather than thinking which oil is good based on the scientific literature that we have studied, based on the lessons taken in school, or based on the what you hear in social media or what you learn from a research paper, both scientific bias as well as personal bias can be overcome when you're using genetic or individualized based approach. So next example I would like to give you is about caffeine metabolism. So everyone, the same dilemma exists in coffee as well, coffee or tea. So if you're asked whether coffee or tea is good, so we will have mixed answers, right? So some will say it's good, some will say limited, some will say have decaf version of it, or some will say black coffee is good. But again, if you're basing it on the profile, genetic profile of the person, it becomes easier without bias how to go about it and how to give recommendation. There are so many bioactives in coffee, but we are more concerned about caffeine here. So if you're looking at the nutritional research, what is happening based on which we are basing our recommendation. So we usually take coffee and we will take a disease condition. So we will learn about the interaction or the association between coffee consumption and a particular disease condition. But here in nutrigenetics, so we are going to see the gene-nutrient interaction. That is, there are different genotypes that exist in the individual. So we are going to learn how these genotype interacts with coffee, whether it will increase the risk or whether it has no effect or whether it decreases. So caffeine, as I said, it's again, there are multiple genes involved in caffeine. So once you drink a cup of coffee now, what happens? It mixes with the blood, it reaches the liver, and it gets broken down into simpler metabolites, and the excess metabolites are excreted in the urine. It's very simple. And when it reaches the liver, this CYP1A2, that is cytochrome protein, is one of the enzyme, which is again given instruction by a gene called as CYP1A2, helps in the breakdown of caffeine to paraxanthine and other metabolites. So this determines the rate at which the caffeine is broken down. So if you are carrying a specific genetic variation, you might be really fast in breaking down. Those individuals, they benefit with coffee. They are called as fast metabolizer. But if you're carrying a genetic variation which makes you to slowly break down the caffeine, then you're called as slow metabolizer, and it takes a longer time for you to metabolize caffeine in your body. So the recommendations will differ based whether you are a slow metabolizer or fast metabolizer. So fast metabolizer are usually the AA genotype carriers, and AC and CC genotype carriers are termed as low metabolizers. So for fast metabolizer, we can recommend up to 400 milligram of caffeine per day, and lean and younger people are likely to metabolize caffeine faster. Whereas when you look at slow metabolizers, we recommend lesser than 200 milligram of caffeine per day. And there are certain foods which will speed up the caffeine metabolism, like grapefruit juice, apples, peppers, and tomatoes, cruciferous vegetables. They are likely to speed up the caffeine metabolism. So rather than saying coffee is good or bad, you're going to base it up on the genetic profile of the person, and then say you're a fast metabolizer, so you can drink coffee, it would be beneficial. Whereas if you find out the person is a slow metabolizer, then you can recommend these given suggestions here. So again, with respect to the diseases that I mentioned now, so we usually relate, right? So one nutrient along with the one nutrient and the disease condition. So we take a population for the study, and we give different the different dosages, like 1 cup, less than 1 cup, 1 cup, 2 to 3 cups, 4 cup. And we identify this particular genotype has an increased risk if they consume more coffee. Right? And here what happens is we are stratifying the population based on the genotype, that is the AA genotype, AC and CC genotype, and based on the dosage. So here if you look at the AA genotype carriers, that people having a AA genotype, even if they have 4 cups, more than 4 cups, they are not at a higher risk for myocardial infarction. It's only the AC and CC genotype carriers, when they are having more than 4 cups per day, they are at a higher risk. So it doesn't mean that more than 4 cups per day is the challenge or is the reason for myocardial infarction, but it is more about the genotype which makes a person susceptible to a higher risk for myocardial consumption on consuming more than 4 cups. So you don't have to really restrict the coffee consumption for these AA genotype carriers. It's only about the AC and CC genotype carriers. You need to ask them to limit the coffee consumption. So it makes a lot of difference. Rather than to say stop it, rather than to say kind of have only 1 cup or have different versions of coffee, you could very well say that it is beneficial for you, whereas it's harmful for you. And same applies to hypertension. hypertension risk as well. So you could see that abstainers 1 to 3 cups and more than 4 cups, actually the risk declines if they have hypertension. So whether it is AC and CC genotype carriers, the more than 4 cups harms only these genotype carriers, whereas it is beneficial for AA genotype carriers. So this example would really make it clear about what we are talking about. How does genetic variation affects the response to the food that we are consuming or the nutrient response. So hopefully I did a honest job in making you clear about what is nutrigenetics, what is the difference, and connecting the dots between genes and nutrients. And at this point, I would like to say that genetics can be used as a tool in dietary assessment in the future. And there are a lot of companies which have started in India, which are doing these genetic testing, and you have a lot of scope in join in these companies as a nutrigenomic counselor or nutrigenetic expert. So if you're looking out to stand out from the rest of the crowd, this is one opportunity for you to delve into it. I'll just quickly talk about sample processing steps. So as I think Sharmila would have mentioned about collection, it gets transported, and collection, it can be saliva or it could be buccal swab, like the cells under your cheek, or it could be blood sample. It gets transported and the DNA gets extracted, genotyping is done, and you get a result to use it for counseling. So these are the 3 types of samples you get. It goes into DNA extraction, and then we call it as next generation sequencing techniques, where the reports are analyzed and you get the results. So this is a snapshot of a genetic report. So you will have a description about the trait that is tested for, and then the results section. Here you could see FADS1 gene followed by the RSID, which is the SNP ID. And your genotype is also mentioned here. Just like your blood report, this is a sample copy of the Nutrigenetic report. So when you read this, you will understand that this person is found to have like a typical levels. So it is enough that he or she ensures consuming a balanced amount of omega-6 and omega-3 levels. So what it is about combining in genotype and phenotype information. So far, if someone is deficient in folate, for example, you would ask them to go for a blood test to find out their serum homocysteine levels, and also to check for their signs and symptoms. And we will also be checking about the dietary intake. You will ask whether the person is vegetarian or non-vegetarian, and we will have a frequency table as well. So now what you're going to do is you're also going to integrate the genetic information along with these 3, so that it will be very helpful for you to get a comprehensive assessment. So likewise, these are ABCDs, right? Anthropometric. I've just mentioned few parameters for you to understand, biomedical parameters and clinical signs and symptoms, and you have different questions on dietary assessment. So now how do we integrate G is you will know about what are the genes that are associated with these traits, and you will be kind of integrating that information along with ABCDs. So we'll have the case study in the next part of the session where you will be learning in depth about it. But I'm just telling you, it has to be along with the current dietary assessment methods, ABCDs, as well as the G should be integrated. So this is how Precision Nutrition Plate looks like for you to get an idea about it. So we have dietary habits, food behavior, and there is deep phenotyping, which is done today, which is made as a mandatory protocol. So no dietitian gives out a diet plan without assessing the biomedical parameters. So in the future, we will be adding nutrigenomics, microbiota, metabolomics, and many more, that is transcriptomics or epigenetics. So once it is commercially available, and all these tools can be used in framing a precision nutrition plate. So it would be similar to your Sabayashi or Leganga, where like you also design it and you can price it really high. And I'm not sure how much impact it would create on the public health, which could be made affordable. More and more people taking these tests, it becomes more affordable. But the whole point is there is a lot of research going on in this field, and we are expecting nutrition to become the core element of healthcare. And Nutrigenetics is all about combined genotyping and phenotyping in nutritional assessment. So thanks again for the opportunity today. And I would come back for the case study session after the quiz. So if you have any queries, you could write to me at journey@instituteofnutrigenetics.in. And this is also our website. You could check. We have different online courses. And we had one intern from your college a very long time back. And it was Ms. Shri Priya coming from there. And we would be happy to accept students for internships as well. We offer a lot of hands-on training. We have a lab in our institute. So anyone for any queries that you have would be happy to get that too. So with this, in my first session, if you have any question, I would like to clarify your questions if you have something. Otherwise, Krishnaveni would be starting off with the quiz session, a short quiz just to ensure that the learning process has happened. Any questions, anyone? I would be happy to answer. Any questions? Okay, let's move on to the quiz. It will be done by Krishnaveni. And after that, we'll do a small case study for you to understand what a genetic report looks like and how we can go about using that genetic report in the dietary assessment. So thank you again.
Good afternoon. So I will be posting a link to the quiz in the chat box. I hope you all can access it. So these are just basic questions. You can answer these within the next 15 minutes. So please ensure that you do it within the next 15 minutes. There are 15 questions, so you will require 1 minute for each. So we are still receiving responses, so I will be closing the form in the next 5 minutes. Please ensure that you finish it before that, and after that we'll have a short discussion on the answers. Okay, so most of you have done a really good job. Thank you for attending, and also thank you for being attentive. So I will be discussing the answers now. I hope you can see my screen. So I'll just go question by question. So the first one is based on the definition that nutrigenetics refers to the effect of genes or genetic variations on the food we consume, which of the following best describes a nutrigenetic relationship? So here, since we are talking about nutrigenetics, so this particularly pertains to how genetic variations that we have impact the food that we consume. So here we have the example of lactose intolerance due to genetic variations in the lactase enzyme gene. So essentially people are not able to digest lactose, which is why they are offered dairy-free alternatives. And the next one is based on the image below, which of the following statements best represents the difference between nutrigenetics and nutrigenomics. So you can see, as the image explains, nutrigenetics basically studies how an individual's genetic makeup affects their response to nutrients, while on the other hand, it's the other way around for nutrigenomics, and it examines how the nutrients influence gene expression. The next one is based on the diagram. How might an individual's dietary choices influence their gene expression differently depending on whether they focus on nutrigenetics or nutrigenomics? So this also underlies the difference between nutrigenetics and nutrigenomics. So the answer is nutrigenetics actually helps individuals understand their genetic predispositions, like the variations and changes that they have in their genome, and how that influences their dietary responses, whereas nutrigenomics focuses on how their gene expression can be modified through the food that they consume. And this is the next question. So this is about a nutritionist who finds a single base pair change in a gene encoding an enzyme involved in folate metabolism, which affects how efficiently folate is processed. So this variation, she found out that is present in 10% in more than 10% of the population, but it does not always lead to noticeable health effects. So based on this information, how should this genetic variation be classified? So here in the image, you can see the difference between a SNP and a mutation. So based on this, how can this diagnosis be classified? So the answer is a single nucleotide polymorphism because it is usually present in more than 1% of a population, and it need not necessarily impact any noticeable health effect. The next one is, which of the following statements is true? So the answer is that nutrigenetics helps personalize dietary recommendations by considering an individual's genetic makeup, which can further influence their nutrient metabolism and dietary needs. The next one is, if the intake of folate can decrease methylation, thus influencing gene expression, is an example of nutrigenomics. Which of the following is another example? So here, if you look at the options, inability to digest milk due to variations in the LCT gene would be an example of nutrigenetics. Similarly, is the third option, which is the inability to quickly metabolize caffeine due to variations in the CYP1A2 gene. That is again an example of nutrigenetics. So the example of nutrigenomics will be the role of butyrate in enhancing gene activation by increasing histone acetylation. This is again an image-based question. So we are trying to understand if this image represents nutrigenetics or nutrigenomics. So as you can see here in the image, these are nothing but genes. So PPAR is something that is involved in adipogenesis. So this talks about how an erratic dietary pattern can negatively impact these genes, the expression of these genes. So this is essentially an example of nutrigenomics. So the next one is based on the image, how do bioactive compounds in food influence health outcomes? So the answer to this is that they alter epigenetic patterns, thus influencing gene expression, favoring a healthy phenotype. So what they can do is they can directly act on the gene which has epigenetic modifications or epigenetic marks imprinted on them. So it has the ability to modify these marks, and then further they favor gene expression, and it leads to a healthy metabolic phenotype. The next one is Mr. A received the results of his neurogenetic test, which revealed the presence of the RS64204224 polymorphism in his BCO1 gene. Based on the image given below, which of the following foods should be included only in moderation? So here the image tells you that the BCO1 gene is responsible for converting provitamin A to active vitamin A. So these are nothing but polymorphisms, the single nucleotide polymorphisms which we have been talking about. So in order to identify them, they are given these RSIDs. So this tells you the effect of these polymorphisms. So if you have these in this particular gene, what happens is there is a decreased conversion of beta carotene to active vitamin A. And if such patients or such individuals consume excess beta carotene from sources like carrot, it can cause the skin to be orange. So essentially what happens is their vitamin A status would still be low because the conversion efficiency is very less. So in such individuals, it is best to include these sources of provitamin A only in moderation. However, you should increase the other sources of active vitamin A, which usually come from the non-vegetarian sources. The next one is based on the image, if a genetic variation is found in the CYP2R1 gene, which of the following statements could be true? So this again tells you the role of different genes in this metabolic pathway. So here we are talking about this one, which is responsible for converting inactive vitamin D3 to the active storage form. So if there is a variation in this gene, this conversion efficiency would not be there, and hence the answer is that the individual is likely to experience symptoms of vitamin D deficiency due to a lack of the active form. The next one is based on the image, if an individual carries the CC genotype for the RS762551 polymorphism, which of the following should they do? So this again is an enzyme that is involved in the caffeine metabolism. So if you carry the CC genotype, you would be a slow metabolizer, which means that caffeine is going to remain in your body for long periods after you consume it, regardless of the amount you consume. So it would result in symptoms like anxiety, palpitation, and nervousness. So such individuals, it is best to restrict the intake of caffeine sources. And then the next one is based on the image provided below. An individual with CRP levels of 7 milligram per deciliter should prioritize the inclusion of which of the following in their diet. So from this table, you can understand that fermented foods, turmeric and fennel seeds are something that act against inflammation, and hence this would be the right answer. Yeah. The next one is Mr. R is an obese individual with a BMI of 31.4 kilogram per meter square. He also experiences frequent episodes of binge eating, which further contribute to weight gain. Based on the image below, which of the following genes should he get tested for? So this image basically tells you the genes that are involved in different functions, which can potentially lead to weight gain. So here the main issue that we need to look at is his episodes of binge eating, which further contribute to weight gain. So if you look at genes that are involved in regulation of food intake and neurotransmitter function, it will help us understand how to address this issue better. So the answer would be DRD2 and MC4R. The next one is which of the following statements best describe the term personalized nutrition? So personalized nutrition considers individual differences, including genetics, gut microbiome profile, lifestyle, and health goals to provide tailored dietary recommendations. The next one is a patient receives a nutrigenetic report indicating a genetic predisposition for lactose intolerance and a higher risk of vitamin D deficiency. So based on this information, which of the following represents the most appropriate critical approach to customizing their dietary recommendations? So the ideal way to look at this would be considering lactose-free daily or plant-based alternatives while confirming vitamin D levels through blood tests before supplementation. So that's the end of it. Thank you.
So hi, coming back to the next session. So what I would like to show you is how a nutrigenetic report looks like and how you will be able to use this in your nutrigenetic counseling. And this will be a kind of an interactive session for half an hour, the next 30 minutes. So I pretty much understand it's post lunch and you're also in a kind of a situation where you want to leave, and we'll make it somewhat interesting, and I want everyone to participate. So here is a whiteboard kept in your hall. I would be giving you a scenario. So all are dietetic students, so you must be knowing like what kind of protocols that goes into diet planning and how do you give a diet plan. So I'm going to give you a situation where you're going to handle a person's diet, and for that you're going to tell me or list down the whatever protocols that you're going to follow. So I'm going to share my screen where the first one would be showing about a report. Okay, so this is a nutrigenetic report, how it looks like. So we have been talking about genetics, nutrigenetics since morning, right? So now we are going to see how exactly. So this is from a company based out of US. So it's a pretty much a good report. I'm not here to promote any of these because you're based in India. And it's not, again, it's not a limitation because these are all done on e-commerce platforms. So once you give your saliva sample, so it is transferred to the lab, and from the lab, they analyze it, and the end report comes into your mailbox. So this is one of a good report, and this Pathway Genomics is a company which has been quite there for a long time. And I thought I'll just show you this personal genetic report. It's just a sample genetic report of a person. So this is how a sample report looks like. So just a preface from the company. And these are how it is framed: personal details, the ethnicity, just like your medical report. And there is a scientific strength rating system, which is kind of a good feature of this report, because you see 4 stars, 3 stars, and 2 star and 1 star given based on the how authentic the SNP chosen is. And then it goes to a kind of a content table of contents like in your book. So they've just mentioned about, so there are about 71 genetic markers tested under diet. So in the diet, you will get to see all these. It's like a summary. And under nutritional needs, there are 9 genetic markers tested. Exercise. So it also talks about genetics and exercise, metabolic health. So here is a summary of all the genetic results that we have got. So if you look at the matching diet type, so it says low carb diet is suitable for this individual. So response to MUFA, response to PUFA, omega-3 and omega-6. So now again, eating behavior traits. So you have all these and the results are mentioned on the right side for this particular person. So you could see easily study, if you study this one page, you will be able to understand about the genetic tendency of that particular person. So you get insights into what type of diet is suitable for that person. So what are the eating behavior traits and what are the food reactions like caffeine metabolism, just like we spoke about. And what are the taste preferences, whether they have lactose intolerance or gluten intolerance. All these can be covered under genetic analysis. And then based on the genetic profile of a person, what would be the nutritional needs? And then metabolic health factors, whether a person will have increased risk for LDL cholesterol or will have enhanced, like reduced level of HDL cholesterol, elevated triglyceride, elevated blood sugar. And it also talks about your body and weight, so how it is. So the next thing is about exercise. So apart from nutrition, you also get insights into exercise, where it tells you what exactly you're suitable for, whether you're fitted for endurance or strength training, what is your muscle power or your injury recovering capacity, and all that is covered under exercise traits. I'll just show you a couple of traits for you to understand. So for example, So here, this is a nice representation where it talks about how you need to distribute your macros together. So for this person, based on the genetic profiling, they have mentioned it as carbs as 40% of their daily total calories, and 30% of their proteins, and 30% from fat. So this is a nice sketch which you could give for a person, because they easily understand a meal plate is usually round, and when you kind of proportion it like this based on the genetic profiling, it's going to be easier. So they also talk about different oils. So this is one is response to MUFA. So you could see the ADIPOQ gene and the PPARG gene, which is actually analyzed, and you get with GC and CC genotype. So for this person, it seems like intake of MUFA seems to have a neutral impact. That is, it is not really beneficial, neither it is harmful, but it seems to have a neutral impact. So this shows that not necessary everyone needs to be benefited with MUFA always. It could be based on the individual again. And if you look at how the diet is like, how their dietary response to PUFA, so this person is kind of having an increased benefit. So the gene tested is PPARG, and the RSID is this particular gene, because you might have many RSIDs or many SNPs under one gene. So that is why they're mentioning the gene, the SNP, and the genotype. So here you can also see a kind of an increased result. So likewise, you could see a description and a gene and a SNP along with the genotype. So this kind of a report is actually taken directly by the consumer, even without a kind of a prescription. We don't need a prescription of a doctor or a dietitian to take. So you can just log on to this company's website. You can just like order for it as you order the other goods in Amazon or Flipkart, and they deliver the sample kit to you. And once you send the samples back, like whether your saliva sample or blood sample, they get it analyzed and they send you the report in the email. So it is all done on e-commerce platform. So it's very much easier. And today's nutritionist, if you're going to learn and get yourself equipped with this nutrigenetics, so it opens a lot of avenues for you in your career aspects as well. So again, coming back to the eating behavior, so you could see that, so you always think like obese people, they have like cravings and they have like a tendency to eat more food, or so it's not just about they are preferring that, but it is also wired in their genetics, where they are actually preferring because they carry certain genetic tendency. Like snacking, it says that it is not associated with extreme snacking behavior. There is a gene called as leptin receptor gene, which is actually contributing, and it is one of the genes which actually makes a person to excessively snack. And hunger, if you look at NMB, and that is again a gene which is tested to regulate appetite and satiety. So here you could see that people with your genotype are more likely to have higher levels of susceptibility to hunger. So likewise, you could also see nutrient needs. It's not just about the diet, macros, and the eating pattern. You could also get insights into how micronutrients, the requirements of a person are. Like it could be the caffeine metabolism, bitter taste, sweet taste, lactose intolerance, alcohol flush. So it's more composite in nature. So we studied about SIP1, A2 in the session. You could see it here along with the RSID, and it shows the AA, which means they're a fast metabolizer. So for this person, you can actually suggest coffee. You don't have to limit coffee unless and until they don't prefer it. So like this, it is a report which will be shared in your group as well after the session. But here, what we would like to do in this session is, how do you use this report in dietary assessment? So suppose you become a dietitian after passing out this degree, and you sit in a clinic, and if someone brings you a genetic report and asks you a question, so, hey, I have taken this report, can you help me to, plan a diet for this person? So you shouldn't be like telling out that you haven't seen a report or you don't know about it, because you should be in a better position to guide others and not really unaware about all these developments happening. And you could also search for, because you are in Chennai, there are a couple of companies in Chennai as well where you could check, go do internships, get trained, and you could also kind of explore whether there's a career opportunity there. So here it is about vitamins and minerals. So if you look at vitamin B2, B6, B12, so it says stay balanced, optimize intake means so you have an increased need for that particular vitamin or mineral based on the genetic tendency. So here, if you look at, so you will see that it's also about the recommendations given based on the genes that are tested. So for example, vitamin B12, again, MTHFR that I told you, right? This is a SNP that they have analyzed for vitamin B12 status, and it says stay balanced. So it has a really smaller impact, but when you go to the other genes, it says that you have to optimize intake because the genetic tendency is contributing to have lower levels of that particular vitamin. So for example, again, this NBF3 is tested for knowing your vitamin B6 status, and this shows that these people are more likely to have lower vitamin B6. So you could actually caution the person that the person might have low levels and to take it. So this is a 50-page report. Similar traits are listed. And you could see it here for metabolic health, for fitness. So all these traits are explained in a similar fashion. So you could go through the report after the session, but I just wanted to show you some examples of it. So now comes the case study. Hope the screen is visible, right? The letters are visible. Yes? Or should I zoom it a little bit? Is it visible?
Yes, ma'am.
Okay. So the case study today, just imagine there is a person there and you have to counsel the person or give a diet plan for this person. I want people to, few of you to volunteer and then come and write it on the board when I ask you to do that. So I'll just first tell you what is the scenario. So Priya is a 23-year-old female. So she works as an analyst in an IT firm in Bangalore. So you could imagine all the IT jobs. So they have night shifts and day shifts. So she works 5 days in a week, and her shift timings are from 7 p.m. to 4 a.m. She reaches home after work at 5 a.m. in the morning. Her breakfast timing is usually between 5:30 to 6:30 a.m. And she has bread or breakfast cereals for breakfast. Then she sleeps at 9 a.m. and she wakes up at 3 p.m. She eats lunch, mainly rice, meat, vegetable, dal, and 1 glass of buttermilk. She has fruit juice in the evening before she leaves. So recently, she has put on about 10 kgs in 6 months and feels difficult to lose weight. She weighs about 98 kg currently, and her height is about 5.7, and her waist circumference is 98 centimeters. She eats dinner at workplace, which is usually pizza and processed foods, and drinks 3 to 4 cups at night, coffee at night, and with 2 tablespoons of sugar to every cup. So that's a good amount when it comes to 4 cups. She's going to get married in 4 months and eagerly looking for a quick fix. Her period cycles are getting irregular, 40 to 45 days. She was suggested about genetic testing by one of her friends. Now help her to reduce weight in a healthy manner by using the information from this genetic test. So now we will be learning in the next couple of minutes, how do you start integrating this genetic information into your diet plan. So here the anthropometric measurements are given. So that's 98 kgs her weight, 5.7 inches her height, and waist circumference is 98 centimeters. Biochemical data is also given here. So I want first one of you to make a nutritional diagnosis of this particular person. So what you will be mentioning in your diagnosis sheet. So what you would diagnose, a nutritional diagnosis first needs to happen. I'll just zoom it a little bit so that you can note it down. What are the biochemical data that are deranged or not within the reference ranges? Have you made a note? So here we have hemoglobin, folate levels, vitamin D levels, cholesterol, lipid profile, iron, calcium, and then uric acid. You have sugar, fasting, as well as postprandial, and all your bun, urea, creatinine, and all that. Okay, so now if you have BCA analysis, so then it also gives you about total body fat percentage, fat-free mass. And then we usually go about the 24-hour diet recall, which is like asked. So now you tell me what all the factors, what would be the nutritional diagnosis, and what would you use to frame a diet plan? What are the factors you would consider? Because I have shown you anthropometry, biomedical parameters, and what are all the other factors, and what would be the nutritional diagnosis? Can one of you just go to the volunteer to go to the board and then just list it down? What are the factors for, that would be the nutritional parameters for nutritional diagnosis? Can you list that quickly, please?
Ma'am, can you hear me, ma'am?
Yes, ma'am. Can I also have a look at the board? I'm just looking at the.
Can you repeat the question once? Can you show the slide? Students are telling it has gone a little fast. They want to read it again, ma'am.
Okay, sure.
Thank you so much, ma'am.
See here, this is the scenario. This is about information about Priya. I want you to do the nutritional diagnosis first. So first, whenever you look at a person, you will diagnose them, right? So this is the scenario. You can just read through for a few minutes and then list down the parameters. So can we have one person to go to the board and then do the nutritional? You just have to list down. So what you have learned from this person and what is the diagnosis that you have made about this person is what I'm asking. Is it okay? Have you seen this?
The board visible, ma'am?
Not, ma'am, not so visible. I mean, if the letters are so small, maybe one of them can read it. Yeah, you go ahead. Whoever is writing can go ahead. Or else, like she can finish it writing on the board and then she can spell it out once she has done.
Okay, ma'am. There's a little doubt from the students' end. You've given the questions and everything.
Yeah.
They're asking whether they have to plan a diet or give the interpretation.
Yeah, so they need to plan a diet at the end, ma'am. But the first question was, what is a nutritional diagnosis? That is what you have learned from the scenario that was given to them. So I have presented the personal information as well as the anthropometric, biomedical, various parameters. So first question is, what is a nutritional diagnosis? That is what they have learned about that person. They just need to sum it up and list it down.
Okay.
The last question would be, of course, to plan a diet. But now the first question, answer the first question. And can one of you spell out what she has written, ma'am? So is the first question done?
Audible, ma'am?
Yes, ma'am.
And the
Can you just
nutritional diagnoses are grade 1 diabetes, grade 1 obesity, PCOD, hormonal imbalance, anemic, vitamin D deficiency, and irregular eating habits, ma'am. These are the nutritional diagnoses, ma'am.
Very nice. And also about the waist circumference, you could see that it's like it's almost 98 centimeters, which also infers about abdominal obesity. So you could also give a diet plan. So first, once a nutritional diagnosis is made, then you could go step by step about how to integrate. So now this you're making it with your anthropometry, BCA, as well as body fat percentage was also higher. You could also include that. And thirdly, you make inferences with your clinical signs and symptoms. So is there any signs and symptoms that you observe from this scenario apart from this? And finally, you could do the dietary assessment. So this is a good attempt But the next thing is, I would also like to show you about the genetic summary. So do you also remember the genetic summary genetic report that I showed you? So this is the genetic report. So you can quickly, we have summarized it here for you. So here it talks about low carbohydrate diet for this person, increased benefit. So can you recollect the what is the gene name here, and what are the RSIDs, and what do you understand by a genotype? So once you know about gene, RSID, and genotype, you will be able to interpret any report. For this, you need to know what are genes, what are RSIDs, and what are genotypes. Hope by this time you're very clear about this. And this is the summary of the genetic report that I showed you. So once you have like a summary, so instead of having 50 pages, you can just have like a sheet or two handy, so that you can mention about these points to your patients when you counsel them for your diet plans. So here it talks about that this person benefits with low carb, and there is an increased benefit from PUFA, and there is a high susceptibility to hunger, fast metabolizer of caffeine, high sensitivity to bitter taste, and they are asking them to this person to optimize intake of B6, B12, B9, A, and highly likely to regain weight because of the associated levels of adiponectin. Because this person have lower levels of adiponectin, they are likely to regain weight. So what would be your recommendation based on the genetic analysis? So now that you have made the nutritional diagnosis based on the scenario, so now I'm asking you, we have also summarized the genetic report. So usually we ask you to do it because of the time constraint. So we have done this, and I'm just sharing it to you. So if you find that the person is a fast metabolizer of caffeine, what would be your recommendation? And if you feel that this person has a higher sensitivity to bitter taste, what would be your recommendations? And if a person want to optimize B6, B12, B9, what are the food sources that you would suggest? So can I have one more volunteer to list down in the board based on this genetic summary? You can take a snapshot or whichever way you want to, and you can just list down on the board. Once you're ready, you can just tell me. So I want someone to give action plans based on the genetic report results. So now that you have got the results of this person, and it says low carb diet is suitable, or they get increased benefit from PUFA, what would be your dietary recommendations for these results? Can you just list down at least 2 or 3 now? Did you understand my question? So screen is here. Can I have one more person who can join? Volunteer someone to write it on the board.
Introducing GoDaddy email.
So is this done? The recommendations? Can one of you spell it out? What's in the whiteboard?
The dietary recommendations are low carb diet, which includes complex carbohydrate, and a high fluid and high protein diet. Fermented foods can be included, and small frequent meals can be included with the correct mindful portion size, and avoid processed and sugary foods, canned foods, and regular exercise should be taken, ma'am.
Okay, so these are like the action plans for specific genes, but once you learn about nutrigenetics, you will also understand there are recommendations that goes for SNPs. So every SNP will have specific nutrient recommendation. I'll just show you how we make a diet plan for a person using a genetic report, because we have quite limited time. I'll just show you my best. How do you do it so that you'll get a fair idea about. So here is the question. So this is how we do it. So first one, when you do the nutritional screening, so you first list down the personal details of the patient. So name, age, female. So this is about the diagnosis initially that is happening. So once you start screening, and then you mention the anthropometric measurements. So the universal screening is first mentioned about the malnutrition screening tool using that. So you mention about the meal preference, consistency of the food, food allergy, dietary restrictions, BMI, weight loss, and then acute disease effect if there is any. Nutritional interpretation goes on. Like this, there's no observed weight loss, no appetite loss, and there is a nutrition assessment note. And biochemical parameters, you highlight the one with abnormal results in red so that the person would know that where they are going wrong and what has to be done to take preventive steps. And then a genetic test report summary goes like this, where we give a sample meal plate according to the genetic tendency, where we bring down fats, protein, and carbs and give them the percentages. And you highlight the points from the genetic report, just as I told you. So they have to increase the vitamin B12 and B6 intake. So we didn't mention about micronutrients now, but you could also mention about choline, copper, lycopene, and CoQ10, which was mentioned in the report. And then limit your salt intake, limit your caffeine, optimize your calcium intake. So these are just highlights from the genetic report. You mention it because we are mentioning only the macros here, but it's also important that we mention about the micros and other parameters which are tested. So when it comes to macros, normal carb, high protein, high fat, and then fats, MUFA, PUFA, saturated fat. So you also get some information about MUFA, PUFA, and saturated. It's not just the fats, but we are also suggesting which type of fats they are better suited for, and just summarize the genetic report. So you just pinpoint, list down the report. So fat storage, it is recommended to reduce dietary fats. PUFA is beneficial, salt sensitivity, gluten satiety, triglyceride levels, and insulin sensitivity is mentioned. And then now you also make some tables like this, which will be helpful to show a lay person if they don't know anything about nutrition or genetics. This will help them if you summarize it. So this is genetics, lifestyle, blood markers. If you have gut microbiome report and food sensitivity, you could also include this. So if you find gluten that has to be limited, see in genetic report what is there, and see if they are eating more of maida or wheat flour based staples. And then if the food sensitivity report mentions something, you can make a column like this to plan out the correlation. So you won't get everything here, you would get only some nutrients here. So what you're doing is a genotype and phenotype correlation. So once you are mentioning about this, the person would know what are the things he needs to restrict and what risks he carries. So what says, like what are the reports that says, according to genetics, he needs to limit the gluten. According to lifestyle, the current dietary pattern says that he or she is consuming more bread for breakfast or any kind of wheat rich or gluten containing food. And food sensitivity report also says this. We have just included gut microbiome and food sensitivity because nowadays these reports are also taken into consideration. So that's why we have mentioned it here. And then finally, you also mentioned about the food sensitivity results, what is actually mentioned in the report, clinical data. So this is like this is the nutritional diagnosis that I wanted you to make. So present complaints, irregular period, past medical history, there's none. And final medical diagnosis is obesity, you said grade 1, and irregular menstruation, that you mentioned it as PCOD. And medications and treatments, none till now. Diet history, so anyways, you will be doing a home recall, 24-hour recall method. So mention that along with the grammages as well as the quantity. So you'll be doing it. Food frequency questionnaire, where we are mentioning about empty calorie. So once you do the nutritional PES statement, so the patient is having irregular menstruation. This is due to higher BMI, and it is evident from the diet recall and the lifestyle choices. So this is what you have done. So now comes the nutrition intervention. So what we are doing is energy, protein, and fat. So we are coming up with the percentage or the calorie calculation based on the Harris-Benedict's equation. And then we are also prescribing diet plan based on the proximate principles. So what has to be the diet plan? The breakup of exchanges is also given. If you don't want the exchanges, you can just avoid it. But I'm just showing you what needs to be done when you are framing a customized gene-based diet plan. So now comes the prescribed menu, where you also take into consideration about what the person is eating, and then you give recommendations based on this, and this is based on the genetic report as well. So whatever genetic insights that you get, you are including it in the diet plan, and also you give additional instructions in addition to the diet plan, like what has to be limited, what has to be avoided. You can give it along with the activity level. So if you feel the person has to take certain supplements, you can also mention this. Hope I'm clear with this. So in case if you have any questions, I would like to listen to you. Any questions, anyone? Hope we were able to give you a fair idea about what is genetics all about, what is nutrigenetics, how does the nutrigenetic report looks like, and how you can integrate the G component, that is genetics, into ABCDs of nutritional assessment. So if you've learned everything, if you're good to go, then thank you again for the opportunity. Thank you, ma'am. And any questions, anyone, you could obviously reach out to us, and my email address is also given, so you can reach out for any queries, any requirements that you have for projects or training. We would be happy to collaborate. Thank you.
Your words of wisdom have inspired us. Also, you have added immense value to our gathering. Thank you, ma'am. Never stop learning because life never stops teaching. The capacity to learn is a gift. The ability to learn is a skill. The willingness to learn is a choice. We extend our heartfelt gratitude for sharing valuable insights and expertise. Thank you, ma'am. Next, Tejaswini from MSc FSMD will be proposing vote of thanks. Very good afternoon to everyone gathered here. Thankfulness is the beginning of gratitude. Gratitude is the completion of thankfulness. Henry Frederick Eliot. First and foremost, I indebted to God Almighty for showering His choices, blessing and being a source of divine light in all our endeavors. It's my privilege to have been asked to propose a vote of thanks on this occasion. We are grateful and thankful to management, Honorable Madam Principal, Dr. Gandhimati Ma'am, and Vice Principal, Dr. Renu Agarwal Ma'am, for encouraging and supporting us to organize international online workshop on decoding the science of nutrigenetics on the topic of fundamentals of genetics and clinical applications of nutrigenetics. On the behalf of the PG Department of Food Science, Nutrition and Dietetics of Srimati Devakundar Nanalal Bhat Vaishnav College for Women, I am grateful to owe my deepest gratitude to our chief guest, Dr. Ferdinand Mary Sharmila, scientist, Jitma Puducherry, and other eminent speaker, Dr. Janani Kamalvanan ma'am, Director of Nutrigenetics. Thank you, ma'am, for taking time to grace the event. Indeed, we are honored by your presence, and all your sessions were very informative and interesting. I express my gratitude to the Dean of New Age Science and Technology, Dr. Subhashni ma'am, and beloved Head of the Department, Dr. Vijayavani ma'am. I would like to thank our teachers and students who guided and supported us throughout the event. I once again thank each and everyone associated with this program. We thank you all for being with us in this event. Thank you. Thank you all once again, and we look forward to welcoming you to yet another engaging and insightful session in the future. Thank you all. This is Roshni.
And this is Shreya Shri signing off together. Thank you.
Thank you.